looked at data about senescent cell dynamics and did perturbation experiments in mice. So this is data we got from Sharpless lab, where there's a marker for senescent cells, P16 promoter, fused to luciferase. So these mice make light in proportion to how many senescent cells they have, or P16 expressed in cells. And you see that the amount right, so this is age, and the y-axis you can think of as senescence. The rise of age very opens up, so you have old mice with 10 times more senescent cells. Some old mice have as many senescent cells as young mice, and other ones have many more. And we got from them the line. So these are individual trajectories of senescent cells. You see senescent cells fluctuate beyond even experimental noise. And that gives a picture like this. And here, I come from physics. This is my natural language. The rate of change of senescent cells is production minus removal plus noise. And using techniques we developed in systems biology, we scanned many equations like that, and basically found one class of equations that can explain this dynamics, and you can kind of make predictions from it, where production of senescent cells rises with age. Why does it rise with age? This could be, this is a pointer. Why does it rise with age? This could be processes like methylation changes, mutations in stem cells that don't kill the stem cell, but make the stem cell produce a lot of damaged cells that become senescent. So you have columns of senescent cells. And this process is basically linear with time. The production rate of senescent cells should rise linearly with time. But the removal, it's not just a constant removal rate, they saturate their own removal. More senescent cells, less removal. And that could be as simple as just overwhelming the macrophages and NK cells that need to remove them, in addition to the 100 grams of tissue they need to remove anyway every day, or something from the sap. So a self-slowing process we infer from the data. And so there's a saturation to the removal. And this together, this rising production and saturating removal, creates an amplifying process where the number of senescent cells, the average number of senescent cells, rises almost exponentially with age. And very importantly,
is beta constant?
Beta in the simplest model is constant, but we can't exclude it also drops with time. Like maybe immunosenescence, we can't exclude that. But did it answer your question? Okay. So, and very importantly, the process has increasing persistence. So if this is the average number of senescence population, some of it, when you're young, it fluctuates quickly, but when you're old, it becomes persistent. Why? Because senescent cells stop their own removal, so they slow down the process. Another individual fluctuates a lot when they're young, but then if they're unlucky enough to be below the average, they stay there for a long time. So this is a persistence that builds up in this process because of the slowing down of removal. And that happened both in the data and in this process. And so this analysis suggested to us that senescent cells turn over very quickly when you're young, with a prediction of about 5 days in young mice, and their removal slows down because they slow down removal in old mice, going to 30 days. That was a prediction from this analysis. So we wanted to test that with Valerie, and the way we did that is induce senescent cells in the lung with bleomycin. After 5 days, you have a lot of senescent cells, and then look what happens with time. If there's no removal, you get those extra cells, senescent cells. Slow removal, fast removal. Then we can tell what the turnover is of senescent cells. And we used the state-of-the-art method that Valerie developed with imaging flow cytometry to measure senescent cells using beta-gal. So the entire lung dissociated and count what fraction is senescent. And what you see is that in young mice, you have about 1% senescent cells in the lung. You induce, and the half-life is about 5 days. And that's just like the prediction from the other data that was separated. So in young mice, there's constant production removal. Every 5 days, senescent cells produce, remove, produce, remove. If you have some injury, you have more senescent cells, they're going to go down. That's why you have these class fluctuations. But in old mice, you do the same experiment, you get induction to about the same level of senescent cells, but it takes them, after a month, they're not back to their basal level, which is 5-fold higher than in young mice. So they have a turnover time of about 30 days. And the agreement between the two experiments, the perturbation experiment in the lung and the luciferase experiment, makes us very confident in the model and parameters. All right, so we have a picture where senescent cells turn over quickly, and then they slow their own production. It's like When you're young, you're a village, you have 100 garbage trucks, those are the immune cells that remove senescent cells, and garbage is quickly and efficiently removed if you have some injury. When you're old, you're at a big city, you're making a lot of garbage, but you're still stuck with those 100 garbage trucks because they're not designed to be old. You have garbage piling up in the streets, a lot of senescent cells. If you have some perturbation, you have more senescent cells, like surgery or something, those garbage trucks are going to take a long time to clear it. If you're lucky to have too little, that's fine. The garbage trucks are working better and you stay there. So it's a way to open up the variation between individuals. And we know that 70-year-olds could have health like 50-year-olds or like 90-year-olds, right? There's a huge variation with age. And so this explains. So now I want to link that, what I asked in the beginning. How can that dynamics be linked to things like incidence rates of diseases and mortality rates? So there's a huge gap between senescence and death, right? There's two different things. We need to make some assumptions. What we don't know in the field is how the amount of senescent cells affects the risk of death. That's an experiment hasn't been done. So we need to make some assumptions. We know senescent cells are removing them helps with many age-related disease models in mice. The way we model it very simply is we say this senescent cell is in one person, senescent cell is in another person. There's a threshold. When you cross that threshold, so that's a mathematical assumption. It could be any sigmoidal function of incidence, but this one we can solve analytically if you like. And when you do that, you find that this stochastic process gives you the survival curve of mice, gives you the Gompertz law.
Wow.
If you add extrinsic mortality, you can also get this. And it gives you a mathematical formula for the slope, the slope, the rate of aging, which we said is so important. And it's basically given by the rate of production of senescent cells divided by the stochastic noise in senescent cell production. So we have a clue about a link between epidemiology and cell biology. And I just want to say that if you compare different species like mice and people, our best estimate is that the rate of production of senescent, that slope with time, is the thing that makes the difference between the aging rate of mice and people. It's about 40 times faster in mice, the rate of production of senescent cells. And that could be due to factors they can discuss if you'd like. What we're working on now is to use that to explain incidence curves of diseases, so different aging-related diseases, hypertension, diabetes, also have an exponential increase in incidence. And the same model, and they drop in old ages, and the same model, we think, can explain those different incidence curves with a different threshold for each disease, and the drop of old age is because all those susceptible have already gotten ill. So you just need two parameters for different diseases. So to summarize, this is a picture, and by the way, I'm finishing early because I want to get your comments, this opportunity, so many experts to get to learn something from me. Senescent cells rapidly turn over in the young, 5 days. They slow down their own production in old mice, and that leads to large and persistent variations between individuals beyond genetics and smoking and stuff like that. Right? There's a stochastic way to get persistent differences, and those persistent differences are enough to explain the Gompertz law and the slope in mortality and link between cellular and epidemiological levels of aging. And also pointing to the immune removal parameter, beta, is an important point of where you can affect the process. Okay, so I'll stop here, and really interested in your thoughts.
First of all, that was great, and thanks God that there are people like you coming to the field, because really, that's another way to understand things for us. So I'll just ask questions from the biological perspective. But first a comment. The bird, the net sharpness method, the P16, is different than the GAU, the value okay? The P16 is being, the problem with P16 that it catches macrophages, this kind of contaminated, our knowledge for now. But my question really is, on one hand, you assume in your equation some linearities. And on the other hand, you show that this is a not linear process. So I don't understand how it works together. And the second question is the NT, how big is the NT? You know?
NT?
Yeah, the NT, how big is it? You
And
know,
what is NT?
the NT,
The production. Eta T. It's not
So,
N.
in the simplest picture, we have senescent cells production rise linearly with age. Like mutation of the stem cells that give rise to more. Senescent cells changes in the chromatin. That's linear. That doesn't give you this exponential kind of rise in senescent cells. But this does. What happens is that senescent cells,
It's non-linear.
or just a few at early times, they remove very effectively. At later times, they slow down their own removal. And that creates a situation where this linear rise turns into say eta t equals beta x over k plus x. And the solution of this is that x is eta t over eta t minus beta. And this thing blows up at a certain age. So you have like and when you can't put in the parameters, you get like, 90 or something. When the senescent cells slow down their own removal so much that they basically can't match the production. And there's a point where production just overwhelms the immune capacity to remove senescent cells, saturates it. And then that gives you this rising amount of senescent cells. And this acceleration is the root cause for the accelerated acceleration you see at old ages, where you fall apart just as fast as a baby develops, and you fall apart in the last baby.
So, first of all, I love the chalk talk, but
Maybe I'm clear.
I guess my question, you're kind of showing, or I'm assuming from what you're showing, that it's mainly clearance, but I'm assuming that the production is it also is getting higher all
Yeah,
the time.
production gets eta t. This is a slope. So this is saying that production rate rises linearly with age. Just a
And
little
it
bit.
might not, but it might not. You still
It might.
have just as much production as decreased clearance, right?
It might not. But this does a great job at putting everything we have. But of course, senescent cells can also augment their own production by what's called bystander effect.
Yes.
Yeah, we don't have to have that in our critical individual. It could be. Removal rate can go down for different reasons, like immunosenescence. So that could, we don't have to have it, but that could definitely, we can exclude it. And the point is that it's a linear process, like the linear methylation changes or linear accumulation of mutations. It's amplified because senescent cells slow their own removal, and you get this catastrophe. And I think every biological process saturates. If you're built to be young and you have a certain amount of immune clearance or injuries when you're young, but when you're old, your body fills up with this injuries. What the package of evolution gave you during your productive years, it just isn't enough. And that creates this universal exponential rise in mortality in all mammals. Same process. Yeah.
So I really like what you did in crossing over to explain the age schedule of death rates. I have a suggestion for you, which I think is a useful way to test this further. So my colleagues and I have looked at mortality schedules for about a dozen different strains of mice.
Yeah.
And they all line up perfectly in association with the reproductive schedule of the strain.
Yeah.
So the strain that goes through reproduction later and has a longer window lives longer. So you've got and I could send you those mortality schedules for each of the strains.
Super.
And all you have to do is look at the senescent cell accumulation in each of those strains. And if those line up perfectly, then you've got a really powerful explanation for why it is that these schedules look the way they do.
Super. And this link between reproduction and life history traits are just a theory for us. Thank you.
It's not a theory anymore.
So yes, I like this. I come from a background in ecology, and there's a ton of modeling in ecology. The most powerful models are those that don't just explain what we already know, but make novel, unique predictions. Have you got some novel predictions?
Yeah, they do. For example, when do you give senolytic treatment? How often do you need to give it? And the answer is you need to give it as often as the lifetime of senescent cells. So if you're worried how often you need to give it, so this shows you that it's useless to give it at young ages because senescent cells go up and down anyway. In middle ages, you need to give it once every 30 days if you're a mouse. Okay? And we think in humans it's probably going to be similar, this turnover time. And we can compute how many senescent cells and how often you need to get rid of and how often in order to rejuvenate it to a given amount of senescent cells, you can say. And the answer is kind of optimistic because you don't need to remove 100%. It's very effective to remove a fraction because you can compute how they go down and then go back up. So it's like having in engineering, you have like Newton's laws. So that's a different prediction. And also all these lifetimes are predictions that we tested in this time. So we're really interested to see the connection between senescent cells and mortality and morbidity in longitudinal studies. That's a missing link for us. We want to understand the differences between senescent cells and different organs and whether different organs are coordinated or not. This central removal process suggests that there will be some coordination because if one organ has more senescent cells, the immune system goes there, and then other organs have less removal and have more senescent cells. So it's like a central process to explain why organs age more or less at similar rates in a given individual. And so there's lots of predictions like that, like very detailed and quantitative ones too.
What happens if
Michal.
assuming the opening step is production of stimulation of senescent cells rather than assuming that the immune system is exhausted?
Good.
תחזור על השאלה בבקשה.
So I'll repeat the question. Here we think that aging timer is the linear increase in production, but there might be a linear decrease in removal. Yeah. So those give you two models which are great candidates to give you rising senescent cells, but just linear reduction and removal doesn't give you the Gompertz law. So it's a way to tell that that's not enough. You need, if you have linear increase in production plus linear decrease in removal, that's okay. We can't exclude that there is, and it's very likely, even though probably the removal of senescent cells is more innate-like, it's macrophages and NK cells, which are kind of like an innate-like part of the adaptive immune system. But the decline of the T cells and the B cells, I'm sure, plays a role. So we can't exclude that. But
T cell and B cell is decline in innate immunity because aging is slow process of innate inflammation.
yeah, so I think that innate immunity is things like the ability of NK cells to remove senescent cells, and they're different in old. Their amounts don't change so much. They're different in old people. They have less granules, but if you do experiments in the mouse and you take the old ones and put them into young mice, they work fine. So there's not something about them is different, but maybe they're exhausted because they're killing so much. And that's this process. What I'm trying to say in a nutshell is certainly there's a decline in removal, but this kind of mathematical approach can help you tease out what's essential, and also maybe get at that rate when we have more accurate data, and then eventually what part of the innate immune system is most declining. So that's, did I answer your question? Partially, but luckily we're on the same campus, so we can continue. Thank you very much.
It's important to make stability analysis. How stable those conditions are. You should actually give a chart for this stability.
Okay, so next speaker is me. This is going to be easy. Just need to stay on time. All right, so I'm going to talk a bit about the other side of the equation, on the immune system side, and I work on trying to understand really immune variation and where it comes from.
It's very disturbing.
I'm a faculty member of the Technion in the Immunology department. And so, as a... So if you want to think about the immune system crash course, this is, the way I think about it is a sensory system that's really about monitoring life and responding to intra or inter perturbations that it sees. So this is really about a sensory system, almost like a sixth sense, if you like, right? And that response happens throughout our lives. And if that's the case, then you can think about the process of going through life or aging as something which the immune system continuously gets exposed. And as we mentioned on senescent cell side, on the immune system side, right, almost every parameter we measure for the immune system has a huge amount of variation in older adults compared to the young individuals. Doesn't really matter what it is measuring. The graphs are usually going to look like this, right? So the old people have a much higher variation than the young individuals. And if you think about where this process comes from, it could be explained by this cumulative hit of going through life and getting exposed. And with every exposure, there is each one of us kind of becomes slightly different, and therefore variation is created. In early life, this may be starting from the genetics, our own genetics. Basically, the immune system, there's about at least 5,000 genes that have directly been associated with immune system function. So even just kind of standard variation between us affects things like cell frequencies. We have other work on this I'm not going to talk about. But then as we just go through life, environment becomes a bigger role in this variation that we see. And in fact, if you take this to the clinic and you measure something as simple as a CBC, a complete blood count, this is probably one of the more common lab tests that are being done, and it's been around since 1957. We've been measuring this as a test. Pretty low resolution for the immune system, essentially 100% of our peripheral blood cells measured at a very low resolution. And if you're a physician, this looks very familiar, and you also are thinking to yourself that it has a limited amount of use. You can kind of see there's some kind of parentheses for what's the normal, not really adjusted for a lot of variation that exists in the population, and hopefully you're not on the extreme side. And every individual you look at really has a very different frequency from one another. But if you look at a person who's relatively healthy coming in one time over the other, they're going to be looking relatively stable in terms of their CBC count on short time periods. The high amount of variation that exists between us on the CBC level doesn't stop there. So if I look, say, at percent lymphocytes, there's at least 100 different types of cells there, if not thousands. And you can see here, there's a group of 30 individuals. Every line here, every yellow line is one person. Looking at the percent lymphocytes, you can see a huge amount of variation and increased variation with age, and also a dramatic shift. And if you look at lymphocytes, they can drop down to a high resolution, and this variation exists at every resolution, and I'm just showing you here CD8 T cell subsets. So there's a huge amount of variation in the immune system at every cellular level you're going to look at. And of course this is important because immune cells have functions and are important to kind of essentially are cardinal to health. And so often when I need to talk about this, I've kind of simplified this to this very simple question a guy walking to a doctor's office would want to ask is basically, hey doc, how's my immune system doing? Right? And so if you think about this from say a cardiovascular perspective, the guy went and said, how's my heart doing? There's a huge amount of assays that the doctor can do, and a huge amount of interventions that they can do to actually both assess cardiovascular health and treat it. But for the immune system, that's really cardinal to health in every kind of single disease. The exception of CBC and a few kind of other for particular diseases, we basically don't understand this, especially if this person is healthy. So we don't really have a metric to assess immune age in any really good quantitative manner, and we don't really understand the variation between us and even the variation that happens in an individual over time, because over time, while at a single kind of short time point, you're going to look relatively stable, each of you is going to be unique. It's almost like a fingerprint, and at a long time span, you're going to look very different from what you looked originally. So it ages. So if we want a metric, the question is how do we establish this for this complex system? And if the system was simple, then getting a metric would be easy. This is a picture of Spud Webb and Manute Bull. People who know them, they used to be basketball players in the late 80s. They're on the same team, short and tall. Very easy to say, here's a metric for something, a single measure. But if the system is really complicated, it's actually quite hard to say what's a good or a bad immune system, how are you progressing along this, and so forth. And so you want to measure this, but as I said, it's complex, and if you try to do this like one measurement at a time, right, like this old Indian parable, people who know this, the blind man and the elephant, I can measure the immune system each from one angle at a time and describe very different things, right? So nobody here would figure out this is an elephant because every person here is looking at a different side of it. Luckily enough, this is no longer a relevant analogy. Basically, this is, I would say, kind of old school measurements of doing single parameters, where new school measurements, the new scientists would be very much high dimensional. So they'd be grabbing the elephant from many, many different angles and measuring them simultaneously in the same individual. And that allows you to start actually quantifying if this is an elephant and how elephants relate to one another, if you like. So the way to do this, especially in a system that's highly variable between individuals, is to get high resolution measurements over time. And this is essentially what we've been doing for the now past 12 years. This is a collaboration with Mark Davis, where I did my postdoc. We started following healthy humans back in 2007. And every year they come to Stanford, we profile their immune system, we vaccinate them with the flu, we collect additional time points, and they come in 1 year after the other. And so this is work we published a few months ago in Nature Medicine, looking at the first 9 years of the study of how individual changes over time. And we're measuring a huge amount of different things on them. For the purpose of this talk, I'm just going to talk about the phenotypes of cells, so high resolution phenotyping of cell subsets, gene expression, and kind of cytokine responses. And so these individuals came in every year, and what we did every year was we were very greedy. We measured everything we could that was cool on the omic front that year. And of course, that was a problem if we wanted to do a longitudinal study. So 6 years into the study, we were lucky enough to have a few samples left in the freezer, and we profiled a bunch of individuals all on a single plate by CyTOF. CyTOF, for those people who are not familiar, is essentially flow cytometry on steroids. You can do 40 proteins on a single cell. Okay, and if you measure these individuals, so I have a subset of people, essentially 15-year-old adults, that I have single plate of 6 years of their data analyzed together. So no technical variation from a measurement side. And then a much larger cohort where I want to be looking at variation, but I somehow need to control for the technical artifacts from 1 year to the other. So let's look 1 second what happens with these 15-year-old adults. So I look at measuring many different cell subsets for them, and this is what you see. So here's one example. Naive CD8 T cells, this is a classic hallmark of immunosenescence, is that naive CD8 T cells drop with age. And in fact, this gray line is the group average slope. You can see naive CD8 T cells, as have been published hundreds of times, drop with age. Okay? But now, because we have longitudinal data for each person, we can, as Uri mentioned, we can measure the rates of individuals, right? And what the rate of changes, and you can see that actually individuals have a very different slope by which their naive CD8 T cells change with time. Okay? And in fact, if I look at this, think about this is not just for naive CD8 T cells, this is happening for every different immune cell that we're seeing and that we're measuring. And so this means if I have a network of cells in a relationship, a stoichiometry of immune cells to one another, they're going to shift over time. And you can see the directions are quite, the slopes are quite different, and sometimes we even get rates going in the direction, the slope is in the other direction, right? So the interesting thing is that there is directionality to the change, right? So we're not just randomly changing from one year to the other. There's a change that's consistent and people are going through it. Okay, so we ask what determines this rate? What determines this change? So the first obvious thing is to look at age, right? So we move to the bigger cohort, normalize it by using the young individuals in the study, so that's the only way we kind of use the young. We sacrifice them for normalization. And we looked at the annual change of the immune cells. So looking here, say in 2007, you can see these individuals, this person came in, had this level of a cell, then he goes up, then he goes down, and so forth. I'm trying not to use slopes to be conservative here, I'm just looking at annual change. And then we're asking, how does the annual change, you go up and down and so forth from one year to the other, for a given cell, correlate with your chronological age, okay? This is a typical plot for naive CD8 T cells, but typical to all cells. We found no significant correlation between the rate of change or the annual change and what your age is, okay? Age has nothing to do with this in older adults, okay? You can see the p-value here, kind of typical. Okay, So that's one point. Is there a direction here? So depending on the types of cells you're looking at, so I can look at this and just plot the distribution of angle change. You can see you have cells like monocytes that have no directionality. Okay? So, okay, this is maybe random noise, except monocytes are highly elevated in old adults compared to young. So that means that sometime before the age of 60, when I started profiling these individuals, their monocytes went up, and at this point they're just fluctuating randomly. Okay, so there's no change anymore, but I got to some level where the monocytes want to be, if you like. On the other side, you have cells like naive CD4 T cells. You can see that the annual change distribution is actually shifted to the right, so it's skewed. So individuals are actually going up every year. Okay, there's a relationship where there's a direction that's clear that naive CD4 T cells want to go up, and I can see this in the course of my study. If you look at this and use it, you can actually leverage this direction, and the fact that you have these individuals sometimes going in the opposite direction, like the different slopes I showed you, you can actually use this information to figure out where the cell wants to be. If it's changing every year, and we have cells like the monocytes that are stopping, we can ask how does that actually happen for each one of the different cells. And so here's a class of cells we call asymptotic, where we can figure out in our data set where the cell wants to be. Why can I figure it out? Because I have the relationship of I have the annual change here on the y-axis. On the x-axis, you have what is my baseline level of the cell, okay, in year one, okay, and you can see, in the year before. So basically, what you can see is a negative relationship. Now, this is what you would expect. The people here are more quantitative would expect this. We did a huge number of different tests to show that this is actually significant beyond what you'd expect randomly and given the constraints here. But basically, what you can see is there's an angle change here, where there's a drop. If you have a high level of the cell, okay, if your baseline level of naive CD4 T cells is going to be high, okay, you're going to drop a lot the following year. If your baseline level is low, you're hardly changing. Okay? In the middle lies zero. What does zero mean? Zero means that I measured you in 1 year, and the next year you had the same frequency of the cell. Basically steady state. Okay? So now I have going up, going down, and steady state in the middle. And if I model this, I can actually figure out where do I cross the line? Okay? Basically, where do I cross the zero? This is basically the attractor point, the area where the system wants to converge. Because if it's higher than this, it's going to drop the system down. And if it's lower than this, it's going to push it up. You can see here the median of the young and the median of the old. It's different from where the point is. This is not regression to the mean. Okay? But rather, for every cell subset in my system, I can try and figure out what the attractor point is. And in fact, that's what we've done. We figure out for every cell where we could identify this with high confidence where the attractor point is. And we put a confidence interval on it. And you can see this for the different cells that we were able to actually identify a significant change between young and old and classify them into 1 of 3 classes. Asymptotic, where I can identify where the attractor point here is, colored in red. Fluctuating, which are like my monocyte cells that have already changed, but I'm not seeing any direction at this point because the changes happened before the age of 60. So they are lying here along some curve of where the system wants to do. And then we have a class we call slow linear, which are cells that are changing, but at a very slow rate, so we can't figure out where they're trying to converge on. Okay? So that's just showing this at a single cell type level. But of course, as I mentioned, we're measuring the entire elephant. So we're measuring this at high dimension, so we can start asking, how does this actually come together? Okay? So if each one of us, I can quantify for you your entire immune system profile by cytoph, and each one of you is a slightly different elephant, right? Then maybe I can take this information and integrate it and figure out what elephant life looks like, right? So basically try and approximate how your immune system ages over time, okay, in one individual. I only have 9 years of data for any one individual, okay? And in fact, by really technologically clean data, we had 4 for this type of assay. And so we took the individuals, all of their different measurements, and we're trying to actually see how they come together. Okay, so could we actually approximate what one individual will look like for their entire life? So to do this, we actually, this is what it looks like. We used an unsupervised trajectory building algorithm called the fusion pseudotime, where every point here is one individual, and in our study in one given time point. Okay, the individuals that are gray are young individuals, and those that are colored cyan or blue are old adults. Okay, and you can see that over time we're starting here with young individuals, and there's very few young old adults, right? But as you shift here, the color scale actually no longer matches chronological age. There's this shift here, where the shift actually, over time, you basically see that the colors are changing, and every point here corresponds to a combination of immune cells for which we can figure whether they're asymptotic, linear, or fluctuating. Okay, so this is the data that goes in. Here's one individual in the study. You can see him here. Basically, his first time point, he looked like this, all the different immune cells. 4 years after, he's here. Okay? So he's shifted. Now, all I did was an unsupervised algorithm. There's no chronological age information going in. All I'm putting in is just the immune cell profiles. You can see this data, basically, I let the computer assemble this trajectory. Question is, does it correspond to anything that has to do with time? Okay, so basically what I'm asking, you can see this person is already positively progressing on this trajectory, right? So it's shifting in a positive manner. We did this for all the different study, all the individuals in the study. In fact, all old adults are moving forward with the trajectory. Every year they're coming into being the blood collecting, their pseudotime, the way we quantify along this trajectory, we call it pseudotime, is moving in a positive manner. Here's one individual, time point 1, 2, 3, 4, and so forth. Okay? So basically this pseudotime, this unsupervised assembly of immune cell profiles corresponds to something like immunological age, because it quantifies your immune system site, and it corresponds to how you age, right, over time. And so we derived essentially a way to estimate this, so we have a way to score this by CyTOF, and we also then developed a gene expression signature that correlates with this immunological age. And the reason we thought this was important is because we wanted to figure out what does this clinically mean, and so the sample size is very small, but we could go to the Framingham study, where there's a huge number of individuals measured, and they have whole blood gene expression measured on the exact same platform we have. Okay, so taking all the clinical information on Framingham, putting it into this framework for every person, we estimate in Framingham what their immunological age is, and include all the other covariates, and we look to see, can we predict survival? And you can see here, people with high versus low immune age actually have significant differences in survival probability. And in fact, the multivariate Cox model is 10 to the minus 4 on this, and the hazard ratio is 1.15 for 5 years. The Framingham study also has methylation data, so we estimated the biological clock, and then we can see that we put them in the exact same system, in the system of equations together. The significance is with the immune age much less than by the methylation age, which is 0.05 versus 10 to the minus 5 for the immunological age for predicting survival. And so basically we have a way, a metric of assessing immune age, and a way of showing that it has clinical meaningfulness, independent of known clinical covariates like age, sex, gender, and diabetes, cardiovascular risk, and so forth. So to conclude, I tried to kind of take this very complex system, and by tracking individuals over time, figure out what is this immunological landscape that we're looking at, what this variation actually corresponds to. Basically, you can think about the immune system as having this psychiometry of different immune cells, right, a different composition. And as we go through life, as I showed you, because of these grades changes, you're shifting the composition of the different cells so they don't look the same over time, okay? And that change is basically what the trajectory, this pattern by which we all follow, we can call immune aging, and this pattern can be now quantified. I can say where I am along this. We have multiple such trajectories depending on other factors, and it has clinical implications because underlying everything that the immune system underlies basically all of medicine, I think. So to conclude, I showed you that older adults' immune system changes in a pattern trajectory that reflects immune aging and is clinically meaningful. It explains heterogeneity in immune profiles of older adults. We can make sense of blood, if you like now, and we can consider this in study design. We can measure it by CyTOF. We now have a smaller panel of flow cytometry that we can assess this with and approximate by gene expression. And I think it opens a quantitative framework to explain human immunity. There may be additional trajectories, factors, identify factors that drive them, and possibly reverse this. With this, I'll conclude. Thank you, and I guess one question is
I appreciate the comparison with the methylation clock, but you have the time to repeat the analysis with rim age, because I think you will get a much more significant p-value for the rim age clock, because it's designed to predict mortality. And the other comment is, as you know better than me, the strongest association between blood cell changes and age pertains to these naive CD8 T cells. And could you just comment on it, because I think you focused a lot on naive CD4 T cells.
So the beginning was naive CD8s. Oh. Actually, the stronger, if you don't scale the information, the strongest driver of the immune age clock is CD28 negative CD8 T cells, which have kind of been, they're roughly speaking, exhausted cells, right? But poor approximate of those. We actually do scale it because we're trying to take into account the complexity of the immune system, and we don't want one dominant factor. I don't think it's a good approximation. We're kind of following the strongest signal. But naive CD8 T cells do play a role. By scaling, we actually take a huge amount of that kind of dynamic range that people report and easy to detect out, and are giving factors to other kind of cell types that are smaller. Naive CD4 T cells change relatively small amounts with age. I said one question, so I'll catch you later. Oh, you're taking your time, okay. Yes, one question.
Exhaustion aging of the immune system is very much associated with elevation of checkpoint, inhibitory checkpoint. Where does it fit in you?
So we haven't looked at this kind of in this system. We've been looking at kind of PD-1 expression on immune cells and so forth. In healthy individuals, it's actually relatively difficult to reliably say that you're measuring PD-1 levels that are meaningful. So if you look at melanoma, immune cells expressing PD-1 in melanoma, huge levels, right? And on the immune system, yes, I see variation, and I think we've published a bit on this corresponding to information, but it's not. I'm hesitant to call it exhaustion. Okay, so without further ado, I'm happy to introduce Michal, who's also from Weizmann, and somewhere here is your... okay, where's you? You can pull me. That's trouble me.
Shai, it was a great introduction to my talk, because there is no one that believes more than me that aging of the immune system is a key factor in aging of the brain. I must admit that I've heard so many lectures lately speaking about how to make our life better, and to have in this meeting the only one who's speaking about Alzheimer, I think I'm surprised, because as we heard yesterday, we can replace heart, kidney, lungs, but definitely we'll never be able to replace our brain. So aging of the brain is a key factor that we need to take care of. So what we know is that aging is a key factor in neurodegenerative diseases, and because of the unique structure of the brain, it was believed, can you hear me? Because the brain is isolated from the blood by barriers collectively known as the blood-brain barrier, it was assumed for decades that the brain cannot be assisted by the immune system for its maintenance and repair. And my team, over the last 20 years, built up a new concept suggesting that the brain needs the immune system for its maintenance and repair. And the narrative that evolved from our study is that the brain is not autonomous, as was believed for decades. And furthermore, the immune system is needed for our mind. And based based on this, we suggested a couple of years ago that boosting the immune system rather than suppressing the immune system can protect the brain and can help fighting against Alzheimer's. So what do we know about Alzheimer's and dementia? We know that it's a disease where misfolded protein are accumulated, such as amyloid plaques and neurofibrillary tangles. We know that the disease is associated with loss of neurons, dysfunction in non-neuronal cells, loss of synapses, local inflammation, collectively leading to cognitive loss. For decades, the Alzheimer's, since it was discovered in 1905, was believed a disease-centric, it's brain-centric, and even neuronal-centric. Over the last few years, it became, we do now that it's also involve non-neuronal cells such as astrocytes, oligodendrocytes, and microglia. What we suggested that the immune system is a player in Alzheimer's. Where are we standing with respect to therapy to Alzheimer's? There were numerous attempts over the last 20 years to fight against Alzheimer's by developing therapy against the amyloid plaques or against the tau pathology, which is accumulation of tau, misfolded protein, aggregated tau. Tau is a microtubule-associated protein that is a key factor in the cytoskeletal protein of the neurons. So all the attempts to develop therapy against Alzheimer's failed collectively. And the question was, where are we? Does the amyloid plaques or the neurofibrillary tangles cause cognitive loss? The answer may be. Does removing them is sufficient? Definitely not. So that means that there are numerous other factors that contribute to disease escalation. In addition, as I said, there is a local inflammation, and numerous attempts worldwide were made to fight Alzheimer's with using anti-inflammatory drugs, steroidal and non-steroidal, completely failed. So where are we standing? Following the last failure in treating with anti-inflammatory drugs, the question was, what are we missing? What we don't understand, even with respect to local inflammation? And I must admit that part of the reason is that the local inflammation was discovered in Alzheimer's. There were many reports in other neurodegenerative diseases that there is a local inflammation, whether it's acute or chronic condition. And again, because of the extrapolation from what we know in multiple sclerosis, attempts were used to anti-inflammation. And the last failure cited our work where we suggested boosting immunity rather than suppressing immunity to fight against Alzheimer's or any inflammatory. So what we suggested after 20 years of working, that the brain needs the immune system for its support. And therefore, using anti-inflammatory drug is like treating frostbite with ice. So it's putting more burden to the brain. So how did we get into this idea? So 20 years ago, we started with a simple assumption that it doesn't make sense that the tissue that is so precious, indispensable, we cannot replace, given up the opportunity to be assisted by the immune system. And my team was the first to suggest that in 1998 and 1999 that the immune system is needed for brain repair. We need macrophages and we need T cells. Subsequently, we discovered that even simple function of the brain cognitive ability is dependent on the immune system. Namely, we suggested that the chronological aging of the immune system is a key factor in aging of the brain. And if you take young mice and intentionally age the immune system, there is a reduction in cognitive performance. So this was the first time that the immune system, the systemic immunity was introduced into the equation of brain function. And what we suggested first, that healing of the brain is like any other wound healing, and maintenance of the brain is like a maintenance of any other tissue, but the communication is different. And it took us several years to discover where the immune and the brain talks to each other. Definitely not through the blood-brain barrier, and we discovered that the main dialogue is between the blood and the CSF barrier. So it's the blood CSF barrier rather than the blood-brain barrier. And this is a barrier that exists between the brain and the fluid in which the brain is floating, which is the blood cerebrospinal fluid barrier. It's the only barrier between the brain and the circulation that it's a epithelial barrier and not endothelial barrier. What we found in this barrier is that there are T cells sitting outside the blood vessels in the stroma of this barrier. And when if there is any injurious condition or pathological condition, immune cells can enter through this barrier. This barrier looks like that. So in this barrier we found this T cell. We found that the majority of these T cells are memory T cells, which means that they are engaged in a dialogue with antigen-presenting cells. Together with Neil Friedman at the Weizmann Institute, we found that the majority of them are memory T cells that recognize CNS antigen or brain antigen. So only paradoxically, the gatekeeper of the brain are CNS-specific autoimmune cells, and they don't cause the disease. So we envision that maybe under pathological condition there is signaling coming from the brain, which we call them SOS signaling, TNF-alpha, IL-1 beta, IL-6, which signal to this epithelial barrier we need help. On the other side, in the stroma, we found this T cell that are memory T cell, and the majority of them are memory T cell, recognizing CNS antigen. Locally, they can produce interferon gamma, IL-4, and IL-10. We envision that the synergy between signaling from the brain and signaling from this T cell can activate this barrier, which is a epithelial barrier, to become a gateway for leukocytes. Indeed, this was the case, and we found a nice correlation between the number of leukocytes in the CSF and the activity of this barrier. Based on this, we formulated the concept that brain fitness is dependent on the immune system, and no matter what we are doing, running, exercises, whatever we eat, affect the immune system, which in turn affect the brain. So the question is, what is the implication for aging and Alzheimer's? So first, we wanted to see what happened with aging to the immune system and how it affects this gateway, the choroid plexus, or the blood CSF barrier. And we found that with aging, there is reduction of interferon gamma-producing cells, which is manifested by elevation of chemokines that are associated with cognitive impairments, such as CCL11, which was reported by a group in Stanford. Based on this, together with Idor Amit at the Weizmann Institute, we wanted to see whether there is a unique signature of aging in this interface, and based on robust RNA sequencing comparing aging of the body and aging of the brain and aging of the interface and young, we found that there is a unique signature in aging of the immune system, which is different from the unique signature of this interface, and we found that this interface is suppressed in aging. Based on this, together with a group in Stanford, we created parabiosis mice, where we connected young mice to old mice, and we found that this aging of this interface is affected by signaling from the brain and signaling from the immune system, again, loss of interferon gamma. We reversed the aging of this interface by neutralizing, using antibody directed to type 1 interferon receptor, and we found that if we take a big cohort of aged mice and we check their cognitive ability, 70% like in human beings, 70% lost their cognitive ability. When we restore the interface between the brain and the immune system, this choroid plexus, we restore the cognitive ability of this 70% of the mice. I'm going through it briefly in order to get to the Alzheimer's. So very based on this, and what was written about this work is that restoring cognition can be achieved by manipulating the place where the immune system meets the brain, which is the choroid plexus epithelium, or what we call it the blood CSF barrier. So what are we proposing? That the functional aging of the brain reflects aging of the immune system rather than the chronological aging, and that's exactly what Shai spoke about. The chronological aging is less a factor, but the aging of the immune system is a factor. So what we are with local inflammation. We don't know what is the chicken and the egg, but eventually they enter into a vicious cycle. Over the years, since we first published that macrophages are needed for brain repair, numerous other studies since 2005 reported that in animal model of Alzheimer's, there is a need for recruitment of blood monocytes into the brain. Remember that in the brain, 10% of the cells are microglia, which are myeloid cells. Nevertheless, it was reported by us and others that macrophages are needed. These are macrophages that we recruited from the brain by creating chimeric mice where all the monocytes are green, so you can visualize that the monocytes are getting to the brain and sitting around plaques. And if you boost the number of macrophages that get to the brain, you can reduce pathology. Genetic study in Alzheimer's, have reported that in patients with Alzheimer's, among the high-risk factors are immune-related factors, again emphasizing the key role of the immune system in Alzheimer's. Independent work by others have already reported that if you create mouse model of Alzheimer's with immune deficiency, the disease onset is earlier and the disease severity is much bigger. So based on this, we assume that aging of the immune system is a factor in Alzheimer's. And indeed, if you go through the literature, you see that in Alzheimer's, what we discovered that the cells that we need are CD4 memory T cell and not CD8. And there are reports showing that in Alzheimer's and in aging, there is elevation of regulatory T cell and a reduction in effector memory T cells that are active, and there is elevation of PD-1 expressing CD4 positive T cells. So taking together, we thought that if we need macrophages to fight against Alzheimer's, if their entry to the brain is dependent on the blood-CSF barrier, let's see what is the fate of this barrier in Alzheimer's. So we took animal model of Alzheimer's in which there are 5 mutations of humans that develop Alzheimer's disease. It's called collectively 5X5. So we took this mouse model and first checked what is the fate of this interface, the blood-CSF barrier or the choroid plexus. The broken lines show you the level of chemokines that can allow trafficking of monocytes to the brain. So this is wild type, and the red showing the Alzheimer's mouse model. And we see that with aging, the level of this chemokine is going down, and the most dramatically is 6CL10, which is a chemokine which is interferon gamma dependent. So in other words, we saw that the trafficking through this choroid plexus epithelium is impaired because the expression of leukocyte trafficking molecules is going down. You can see it by mRNA, and you can see it by immunostaining. We checked to see what might be the reason. So first we got human sample in which we found the same phenomena, reduction of expression of trafficking molecules by the choroid plexus with aging and in Alzheimer's in patients. What we found that in this mouse model, there is reduction in interferon gamma producing cells with the disease progression and elevation of regulatory T cells, together telling us that in this mouse model, there is reduction in interferon gamma producing cells and more immune suppression as expected in aging. So it seems that in this mouse model, there is accelerated aging. Independent work, which was published by a group in Chicago, found the same phenomena in different mouse models, which is J20. Again, reduction in interferon gamma producing cells and reduction in expression of trafficking molecules by the choroid plexus. So what can we do? How can we fix it? So there are 2 options, either reducing the level of suppressor cells or boosting the level of effector cells. We give the 2 options. I'll give you a brief here and continue with the immune checkpoint. So we'll create chimeric mice where we can reduce which allow us to reduce suppressor cells, and we found that following the reduction of suppressor cells, FOXP3 regulatory T cells, those of you who are immunologists here, and we found that we improved significantly the cognitive ability a month after the treatment. This is a maze where the animals are introduced into a pool of of water, there are pictures around the pool, there is a platform that the animal don't see, but if they touch the platform, they can look around and learn the position of the platform. So Alzheimer mice don't learn over the maze. They are the red, black, wild-type animal, normal, healthy, they learn. And the blue show you cognitive ability of mice that are Alzheimer mice in which we depleted regulatory T cells. Based on this, we envision that maybe immune checkpoint blockade will have a similar effect. Those of you who are not familiar with the immune checkpoint, they were discovered by the group on Onjo in Japan, for which he got a Nobel Prize this year. Basically, we're speaking about program-based protein, that the level of which goes up with aging. When the cells are exhausted, the more you use them, the higher the level. And if you block either the receptor or the ligand with antibody, you can boost the level of effector memory T cell, either CD4 or CD8. We envision, because of the stroma of the blood CSF barrier, there are epithelial cells, regulatory T cells, and antigen-presenting cells, all of which express the ligand. We thought that if we give either an antibody to neutralize the receptor or the ligand, we will have a higher level of CD4 positive T cell that will produce interferon gamma. In other words, the idea is to rejuvenate the immune system by giving antibody that will block either the receptor or the ligand. We did so, and we gave the animal antibody directed to PD-1. We saw that there is elevation of interferon gamma, and we saw that there is elevation of trafficking molecule by the choroid plexus, and if we block with anti-interferon gamma, I just started the checkpoint. So as a result of the treatment, we saw that there is improvement of cognitive ability in the animal model of Alzheimer. This is a maze where the animal, we measure the number of errors that the animal do before they go into the right platform, into the maze, into the arm with the platform. The red show you wild-type, the Alzheimer animal. The black show you normal animal. The gray show you the placebo, and the green show you anti-PD-1 treatment. We saw that there is a reduction in the plaques as a result of the treatment. Also, the treatment was not directed at all to the pathology in the brain. When we treat it early, we see that we prevent loss of cognitive ability. The red show you Alzheimer mice, the green show you Alzheimer mice treatment with anti-PD-1. We repeated it with anti-PD-L1 because PD-1 can recognize both PD-L1 and PD-L2. We did a dose dependency, and you can see that the low dose is ineffective, the high dose is effective, and animals receiving anti-PD-L1 behave very similar to animal, the wild-type animal. Amazingly, because we wanted to see whether we reverse, we took a cohort of mice, measured cognitive ability, so that we completely lost, and then divided to two groups. One received the placebo and one the anti-PD-L1 with reverse cognitive loss. Because the treatment is not directed to the amyloid plaque, I'm finishing. Because the treatment is not directed to the amyloid plaque, we wanted to see whether it will be a general phenomenon. So we took our animal with dementia, where the cognition is impaired, but it's a short-term memory, it's a tau pathology. In this animal model, you measure short-term memory, so it's a maze with 3 arms. First, you habituate the animal to learn the two arms, and you don't open the third one. If they learn and remember, they will have preference to the third arm once you open it. So wild-type animals show clear preference to the novel arm. Alzheimer mice, no preference. When we treat with anti-PD-L1, we restore cognitive ability to the extent of the wild-type animal. You can see it very nicely here. This is the behavior improved with the dosing. We reduce the inflammation. We reduce also the level of aggregated tau. So in other words, we reverse the disease and slow down the disease progression. We are not reviving dead neurons, but we rescue neurons. So what is the underlying mechanism? And I'm finishing with it. What we found is, as a result of the treatment, there is recruitment of monocytes to the brain. we found that by single-cell RNA-seq, that these macrophages express, first of all, we found that there is recruitment of monocytes. When we block the recruitment of monocytes, we completely lost the beneficial effect of the treatment. And together with idoamide, by single-cell RNA-seq, we were able to show that the macrophages that get to the brain express many scavenger receptors that are not expressed by the microglia. So to summarize, what we are proposing, that immunotherapy is a way to rejuvenate the immune system in order to facilitate curing the brain. We don't think that we are curing Alzheimer's, but we believe that it's a way to stop disease progression and turn it into chronic disease. Importantly, we are using immune checkpoint blockade, but based on the mechanism, a single injection has an effect that lasts for 6 to 8 weeks, so it will be intermittent treatment rather than chronic treatment. And based on what is needed for Alzheimer's, it will be a treatment with less adverse immunological effect. This work was heavily supported by the ERC. There are numerous graduate students and postdocs who participated in the work. All the single-cell RNA-seq with a tight collaboration with Professor idoamide. And since it's a 20 minutes of talk of 20 years of work, sorry for taking more time. And these are the people that contribute to this work.
We just need to be on schedule, so please take a break. Be here at 10:30. The faculty, please stay here.
And various diseases of aging, and the precise biology of this is not fully understood, and we're still missing good biomarkers of mitochondrial function and mitochondrial activity, but we know that the mitochondria is involved in the pathogenesis of Alzheimer's, diabetes, cancer, and the entire process involved in aging. Let me use this movie, if I can, to tell you about how in our bodies, of course, we have multiple cells that contain a number of organelles, as I mentioned, but importantly, the mitochondria, which contain these chromosomes that I mentioned, that have 13 large protein-coding genes, 2 ribosomal RNAs, and this was thought to be its main genetic function. But over the last 15 years, we've identified multiple small open reading frames, 2 of which involve a peptide called humanin, which comes out of the 16S ribosomal RNA. Humanin is a neuroprotective molecule that's involved in longevity. Its administration increases lifespan in various organisms. And a second molecule known as MOTC, coming from the 12S region, which I'll speak about a little bit more, is also secreted from the mitochondria, gets out of the cells, goes into the systemic circulation, and these 2 and several other mitochondrial peptides have important protective actions on liver, heart, and importantly, brain. So, beyond the importance of the mitochondria itself, I want to talk to you about micropeptides. And you know, of course, that the genome has over 3 billion nucleotides, but we were surprised to note that only 20,000 large protein-coding genes are found in the genome. In the mitochondria, which has this very small 17,000 base pairs in this chromosome, there were 13 protein-coding genes, remarkably few for this complex organelle. The next level, the next revolution in genomics over the last decade has been the recognition of the importance of RNA molecules. Various non-coding RNAs, and there's more of those than there are genes. The mitochondria, in fact, has over 1,000 RNA species with a variety of activities. But the next revolution in genomics I propose to you is what I call the micropeptide era. And there are millions of micropeptides that come from the nucleus and close to 1,000 that come from mitochondria. And these, I believe, are important, particularly in diseases of aging. And each of these waves of the genome has brought with it a variety of precision medicine and other approaches to treat disease. And here you can see that the growth in the number of these micropeptides has been evolutionarily dependent, with more of them appearing in higher organisms. And in fact, the latest discovery just last week in a cell paper from a group at Stanford indicated that the microbiome in bacteria contain thousands of micropeptides that may generate many of the beneficial effects of the microbiome. But the mitochondria has its own micropeptidome, which I believe has critical roles in influencing diseases of aging and potential treatments for these diseases. So my lab is involved in developing this concept of the omics, or the system biology of the mitochondria. We look at these mitochondrial peptides in terms of their structure, function, and signaling. We've been involved in defining what these genomics of the mitochondrial orpheome are. We are interested in harnessing mitochondrial transcriptomics, as I mentioned in one slide. The epigenetics of the mitochondria. There are, in fact, epigenome modifications of the mitochondrial DNA. We're trying to understand peptidomics and mass spec identification of these peptides, and the metabolomics involved in this. But I want to share with you today particular discoveries that we've made relevant to population genetics of mitochondrial DNA, mitochondrial peptides, and how that led us to identify potential novel approaches to dealing with diseases of aging. We've also developed a variety of assays for mitochondrial peptides that could be important diagnostic and prognostic tools. And finally, of course, these peptides could prove important targets in drug development. So our discovery pipeline in the lab starts with looking at the genomics, and we do an exercise that's essentially identical to what's known as GWAS, which is a process that has traditionally ignored mitochondrial DNA precisely because the mitochondria is only maternally inherited and does not obey Hardy-Weinberg disequilibrium. And all of the GWAS studies that have been published until recently eliminated mitochondrial DNA from their analysis. And I'll show you some discoveries that we've made there. Similarly, when people look at RNA-seq, they throw away mitochondrial data, or maybe just look at the 13 mitochondrial large genes. We've developed an approach that allows us to see changes in the specific expression of mitochondrial ORFs. We're trying to now identify these in terms of mass spec peptidomics. Then we take these peptides and bring them back to traditional biological studies in cell line and mice with the goal of developing potential drugs, at least one of which is already being developed, as I'll mention in a second. So a couple words on this technique that we call MIWAS, which is not about me, it's the GWAS of the mitochondrial. And we've used the definition of these mitochondrial ORFs to see if we can find particular changes in the mitochondrial DNA that's associated with disease. And what we do is we look at the mitochondrial DNA and look for SNPs that have specific effects on changing the amino acid composition of these theoretical ORFs, where a change in this mitochondrial peptide might lead to altered biology. And I'll show you two interesting examples of this. So basically what we're doing is that we've gone into a variety of existing studies that have conducted GWAS analysis and identified genes involved in disease and other traits, and went back and looked at that unharvested mitochondrial data. And here are two examples that are unpublished. On the right, you can see a particularly exciting discovery that one of my graduate students, Brendan Miller, has made, where he found a SNP in the loop, D-loop of the mitochondria, that carriers of this SNP have a 5-fold increase in the rate of cataracts. And while that may not be a disease that kills you, It is probably one of the most common diseases of aging, and clearly the people who have this SNP have a dramatic increase. Interestingly, only people of Hispanic background carry this SNP. On the left, you can see another NIWAS analysis we've done on patients with colon cancer, and we've identified a SNP that actually protects people from getting colon cancer, and this SNP is specific to African Americans, which, by the way, have an increased risk of colon cancer, but those who have this SNP have a reduced risk. So these are, incredible opportunities to try to understand these diseases from a new perspective. But the disease that I've been connected to my entire career, I started doing work in diabetes and the Technion together with Nir Barzilai and Eddie Carnielli, literally almost 40 years ago. And I've been very interested watching the rise in the incidence of type 2 diabetes in the United States, which used to be unheard of in children, and now we can see that in certain ethnic groups, type 2 diabetes is significantly appearing in children of certain ethnicities. Most affected are Native American and so-called Mexican Americans. And there's a particular observation that's probably 20 years old that you can see here, that in Pima Indians, some of you may have heard of Pima Indians, they have the highest rate of diabetes in the world. The majority of adults in this tribe have diabetes, and you can see that people who have a diabetic mom have almost full penetrance, and they also have diabetes, which should have made somebody think that maybe there's a mitochondrial DNA inheritance involved in this. So we went to something called the Health and Retirement Study, which is run by a woman called Elaine Kulis, who's one of my colleagues, and used that GWAS database to try to find new SNPs that might confer risk for diabetes. And we've identified a SNP in the mitochondrial DNA that's associated with increased hemoglobin A1C. And lo and behold, this SNP is again unique to what might be called Hispanics, but really they're Americans. They're either Native Americans or Mexican Americans or Central Americans. You can see in green the frequency of this gene variant is between 20 to 50% in people native to America. People who carry this SNP have a threefold increase of diabetes, as well as higher hemoglobin A1C, and we've validated this now in 3 different cohorts, all of which show that Hispanic who carry this SNP have an increased risk of diabetes. So what does this SNP do? Interestingly enough, this SNP does not change the sequence of the amino acids in the parent gene, known as ND2. It's one of the respiratory chain proteins that comes from mitochondria. But what the SNP does, it eliminates a start codon in a novel open reading frame that encodes a peptide that was previously unrecognized to be important. We call this peptide MENCH, and we encode in NA270 human. I like to give these peptides colorful names. And this is a novel mitochondrial peptide hormone absent in 30% of Americans that we think could be an important tool in understanding, diagnosing, and treating the diabetes that is afflicting these people. You can see here that when we inject this peptide into mice on a high-fat diet, we improve their glucose tolerance, we improve their blood glucose, and we developed an analog of MENCH, that we call MENCH32, which has a dramatic effect in preventing weight gain on a high-fat diet in several mouse strains, both diet-induced obesity and genetic obesity. And this is entirely a result of loss of fat, and there's no change in muscle mass. And there are many other interesting effects of MENCH in rodent models. It lowers insulin levels, improves insulin sensitivity, improves blood sugar, body composition, but also has effects on blood lipids, insulin secretion, and remarkably, it lowers glucagon levels, which is a factor that has major pathogenic effects in diabetes. And the effects, while the mechanism for MENCH action is not fully understood yet, the effects clearly have involve fat metabolism. You can see weight loss in OB mice that's associated with a very specific metabolomic signature, which is associated with a transition from burning carbs to burning fat, and a simultaneously dramatic increase in spontaneous activity in these mice. So really, with the rise in the popularity of various diets you're going to hear about from Walter Longo, my colleague, later today, we think of this molecule as a ketomimetic peptide. It transitions the body to a metabolism that one sees in a ketogenic diet. And we can also see this effect of Mensch on fat cells in vitro. And remarkably, when we look back into the healthy retirement study and other studies, carriers of this SNP are also heavier. So it's essentially a thrifty gene that helps people conserve fat in times of limited caloric intake, but has become a major liability in the world of excess calories. So it's an exciting peptide, and I think there might be both therapeutic and diagnostic potential there. But I want to switch now to the role of the environment, which is really the title of the session. And I really think that the secret to long life is diet and exercise. I should do a better job on both, like most of us. But I want to argue that a lot of the effects of these healthy lifestyles is through regulation of mitochondria and mitochondrial peptides. We described the first mitochondrial peptide, the first really important micropeptide in my mind, which is humanin, that is very diet dependent. I like to think of it as fasting mimetic peptide, but I'm not going to talk about today. I'd like to share with you some new observations of this peptide that we call MOTSI, like motzi lechem in haaretz, but it's MOTSI, which is very exercise dependent. And you know, I like to call my bathroom gym, so I can tell people I go to the gym first thing in the morning. But really, its exercise is critical. And MOTSI, which we published in 2016, just 4 years ago, is a peptide that comes from the 12S ribosomal RNA region. David Lee, which was a student with Walter Longo and a postdoc in my lab, led this effort, and over the last almost decade, we've created a body of work demonstrating that MOTSI is a peptide that has very potent effects on mice with a high-fat diet, or obese mice, leading again to tremendous weight loss. It works mainly by activating AMPK and has this remarkable effect that when you inject it into mice that are high-fat diet, not only do you see a reduction in weight, you see almost a complete elimination of fat in the liver, and there's no effects on mice who eat a regular diet. And as a result of this observation, MOTSI was licensed to a company that I co-founded with Nir Barzilai called Kobar, which is currently developing it for NASH and other indication, and it's in phase 1 clinical trials. And I argue to you that MOTSI is an exercise mimetic and an exercise promoting peptide. You can see here in work that's now submitted that both acute exercise on the right causes an immediate rise in circulating levels of MOTSI, and chronic exercise, a 3-month trial of exercise, causes more than a doubling of MOTSI in people who were previously sedentary. Furthermore, again, work done in collaboration with David Lee at USC, who is now about to become an associate professor there, mice, both young and old mice, that are treated with MOTSI daily, you can see that their ability to stay on the wheel in these mouse treadmills is dramatically enhanced by MOTSI administration. And this effect that you can see in old mice is particularly effective, allowing old mice to run on wheels almost as well as young mice, in terms of number of hours a day that they can do that, or seconds before falling off. One of the mechanisms we think is involved in MOTSI's activity is that it suppresses myostatin and also changes the ratio between fast and slow twitch fibers in muscle. And even more interesting is that this may be a different effect in various ethnicities. And we've been very interested in how a variant in this might affect elite athletes. And in collaboration with a group in Japan, we've identified a SNP in MOTS-C that is found in 8% of Japanese, Koreans, and Northern Chinese. And that's a lot of people, by the way. We're talking about hundreds of millions of people. And this SNP causes a conformational change in the MOTS-C structure. And on the other hand, the SNP is, of course, part of the 12S ribosomal RNA. And you can see that little red arrow on the right. It essentially does not change the structure of the ribosomal, the mitochondrial ribosomal molecule. So what is the effect of this change in MOTS-C? So first of all, from an epidemiological perspective, you can see that Japanese people, and this is meta-analysis of 3 different groups, total of over 10,000 patients, people who carry the SNP have almost a 40% increase in the risk of developing diabetes. And even more interesting is that in one of the studies that everybody also had one of these Fitbits, you can see that the effect of the negative effect of this SNP is only in sedentary people. So in times past, where everybody was active, this would not have been a bad SNP to have. Today, where so many people are sedentary, this is a major increase in diabetic risk. And another very interesting effect here is that this is only true for males. We didn't see it in premenopausal women, but in mice experiments, we can see that an ovarectomy of female mice makes them again responsive to the MOTS-C effect. And here we show you that the mutant peptide, which we call K14Q MOTS-C, is essentially inactive. So the normal peptide that everybody in this room, unless there's a Japanese person that I missed, everybody in this room makes the normal peptide, which leads to weight loss, improved glucose tolerance, and in vitro improvement in insulin sensitivity of muscle. But the abnormal peptide fails to do that. So there are millions of people in Asia who don't make this peptide, and to me this is a compelling opportunity for a precision medicine approach to this type of diabetes. And the last thing I want to tell you about MOTS-C is that it does in fact fall with aging, and the levels are lower in old folks versus young people. This is in mice, but it's also true with humans. And work done by a postdoc at David Lee's lab in collaboration with us. You can see that administering MOTS-C to mice starting in midlife 3 times a week, which is obviously underdosing this, results in an increase in both median lifespan and an insignificant increase in maximum lifespan. I think that if we had the opportunity to do long-term treatment with daily injection, we could get a substantial increase in lifespan and obviously in health span. So just a word on the other omic system biology approach that you use, which we call MDP-seq, or microtranscriptomics. We can analyze RNA-seq data sets and see that there are specific changes in open reading frame expression in a variety of disease, again, opening the door for novel discovery, potential peptides that might be important in various diseases. And I'll finish off by summarizing that the normal mitochondria makes multiple peptides, and there are several more exciting peptides that I love to talk about, but I also have 20 years of work to report and only 25 minutes. So all of these normal peptides support the health of the body and affect multiple tissues and protect from NASH, obesity, diabetes, as well as other diseases ranging from eye disease to cancer, and really an exciting opportunity for advancing the science of mitochondria and aging. I want to thank many of my collaborators, several of which are in the room, Nir, Walter, David, thank you for your support. The tremendous people who've been working and helping in my lab, a lot of the funding agency, including AFAR, who've been instrumental to my career. And I just want to say we've created tremendous amount of resources and reagents for mice, antibody peptides. And anybody who has a thought, just shoot me an email, and I make these available to the community. So thank you.
Phenotype of mitochondria in the age similar to the phenotype of mitochondria in the young under inflammation?
That's a great question, which I don't have a precise answer for, only to say that during aging, there is a dramatic change in mitochondrial phenotypes, both in terms of proteome, transcriptome, physical appearance in microscopy, and actual function. Whether inflammation mimics aging, specifically, maybe there's literature on that, but if not, I'll come back next week and we'll do the experiment.
Do you have a sense for which, if any, of the peptides are induced by metformin treatment in a mouse organ?
We're actually doing this right now using the microtranscriptomic.
Can you repeat the question, please?
So the question is specifically regarding metformin. And those of you very interested in metformin, just like Nir taught me that it's important, that there are probably 18 different potential mechanisms of action for metformin, several involved in mitochondria. And the question is, could metformin be inducing a mitochondrial peptide that mediates part of its effect? We've asked this question. We've approached it from all of our omics approaches. We actually have a couple of candidate peptides that we're testing now. There's an RFA in January that we're eyeing. And I want to thank all the people in the audience, that includes you, Nir, who deposited their RNA-seq of metformin treatment that allowed people like me to probe. So maybe next year, if we're going to do this again, I'll have data to show.
How about NAD plus boosters, such as nicotinamide, riboside, or others?
What did they do to these peptides? Each of these is a fantastic question, and people who have systems that they want to ask these questions around, I'm happy to collaborate, but I don't have an answer to that.
Yeah. Thank you for the talk. You made my day. And
you just made my day.
So it's mutual. So I wonder if you can care to comment on the relation between the mitochondria and the gut microbiome.
So that's a great question, which I've also pondered. As you know, the mitochondria is really a bacteria that entered a symbiotic relationship with the eukaryotic cell, I don't know, a billion years ago. And we've looked at the open reading frames in bacteria. I mentioned this group in Stanford who completely scooped us. What I can tell you is that there's almost no homology between bacterial micropeptides and human mitochondrial micropeptides. But at the same time, there seems to be crosstalk between
like signaling
from both. And again, David Lee, who worked on MOTS-c, has a paper in development showing that MOTS-c directly affects bacterial function, and also actually my daughter who worked in this lab showed that human affects bacterial function, and there probably, we haven't tested this yet, bacterial micropeptides are very likely to affect mitochondrial function. These are all great questions that require many millions of dollars and a lot of hands to answer, but I think are worth looking into.
Good question. Very nice talk. Let me try to see whether, because diabetes is one of the risk factors in Alzheimer's, the biggest risk factor, let me try to see whether you have any cross between.
Great question, and the answer is absolutely yes. And several of the peptides, for example, humanin, which Nir and I worked on 20 years now, is beneficial both for diabetes and insulin resistance and protects against neurodegeneration in various models. And Gil Atzmon is here, and we've worked with Gil and Nir to identify a SNP in humanin, which seems to protect against Alzheimer's disease, induce longevity, and also have probably roles in preventing insulin resistance. So absolutely, there's a lot of overlap. And I feel like I'm stealing time from the next speaker. All right, thank you.
For discussion, I'd like to call Rafa De Cabo. Rafa is a senior investigator and branch chief at the National Institute
of adults with obesity. So a lot of people that we're looking at. We know that obesity dose-dependently increases the incidence of diseases that are commonly associated with aging. So a really good example here is arthritis. This is the percent of people with arthritis or any of these diseases. You can see that compared to people of normal weight in this dark blue line, people with a body mass index in the overweight range, they have about a 50% increase in the incidence of osteoarthritis, and people with obesity have a more than double incidence of the osteoarthritis. So, you know, a good way to promote age-related diseases is to perhaps be having extra weight. The same is true of diabetes, where you've got like a tripling of the incidence with overweight, and with obesity, it's like a 5 times greater incidence of diabetes, and similar for other diseases. So excess weight is associated with diseases of aging. What do we know about weight loss? Because these are different questions. Just because obesity is associated with a disease doesn't necessarily mean that weight loss is going to reduce that disease. But now there have been relatively large-scale studies, and there has been a meta-analysis which has shown that weight loss interventions that are deliberate interventions, not just accidentally losing weight, they do seem to reduce the risk of mortality from diseases related to aging. So, for example, all-cause mortality is reduced by intentional weight loss. Also, incidence of cancer seems to be reduced. Heart disease seems to be reduced. As Volta said, with the monkeys, you know, you might have a lower incidence of mortality from various diseases, but sooner or later you've got to die one day. I mean, we've all got to die. Maybe some of us here taking metformin might defeat that rule, but sooner or later we've got to die. But what is the quality of life? And that's where looking at the effects of weight loss on health outcomes, I think, is very important. We know that in terms of weight loss, and one thing that I should mention is that in these studies that were analysed in this meta-analysis, the weight loss interventions were predominantly food-based interventions based on eating a low-fat, healthy diet. However, we also know that food-based diets don't tend to induce large weight losses. And we do know now that the greater weight loss that is achieved, the greater the health benefits. One example here from an increasing number of examples is osteoarthritis. Now, in this table, it splits up people who had lost weight with a diet program, a partial meal replacement program, and then it retrospectively split up the participants in the study. There were a few thousand of them into people who lost less than 2.5% of their weight, 2 to 5% of their weight, 5 to 7, 7 plus and 10 plus. And what they found was that when people lost 7.7% or more of their body weight compared to their baseline body weight, they had a clinically significant reduction in self-reported pain from osteoarthritis. So a 5% weight loss wasn't enough, didn't make any difference to participants in how much the pain bothered them. It was the 7.5% or more weight loss that resulted in that clinically significant benefit. Here's another example from a similar kind of study. It was also using a 12 to 16 week meal replacement diet, and they asked people how much pain they felt in their body before and after the diet, so pain from any source. And on the left here we have people who lost greater than 10% of their body weight during the diet, and on the right we have people who lost less than 10% of their body weight. And the size of these dots represent how many people reported that phenomenon. So a big dot means a lot of people saying, yeah, I felt that too. And the colour of the dot represents, so blue represents less pain and red represents more pain. And you can see that it's the people who lost more than 10% of their weight who had these big blue dots on their bodies, meaning that in those parts of their bodies, they felt, a large number of people felt a reduction in pain. But it was related to 10% or more weight loss. Similarly, there's very exciting recent data for diabetes remission. Here's a study from the UK where people were put on a lifestyle 12 to 20 week total meal replacement diet for 12 to 20 weeks. And at the end of 1 and 2 years, they measured how many of these people still had the symptoms of diabetes, or the signs of diabetes. Everyone at baseline had type 2 diabetes. And here you have the people who lost less than 5% of their body weight, or less than 5 kilos, 5 to 10 kilos, all the way up to people who lost 15 kilos or more of their body weight as a result of that intervention. And what is phenomenal is that there's this really dose-dependent curve, this dose-response curve, in terms of the proportion of people who were diabetes-free at the end of either 1 year in orange or at the end of 2 years in red. And phenomenally, 86% of people after 1 year who had diabetes no longer had diabetes 1 year after a weight loss program that induced 15 kilos or more weight loss. And even the effect persisted at 2 years. So the more weight a person loses, if that person has overweight or obesity, the better. In weight management, there used to be a common saying, I guess that was in the old days when people didn't have so much excess weight. A common saying was, oh, if you lose 5% of your body weight, you'll have health benefits. Just go away and lose 5% of your body weight. Well, it's true. There are dose-dependent benefits of weight loss, and 5% weight loss will bring some benefits, but it's not enough to bring those really clinically significant results. So I say, when it comes to weight loss, 10% is like the new 5%. We need those big, beefy weight losses. The most effective way to lose weight, besides bariatric surgery, which is extremely effective, or besides obesity pharmacotherapy, which is also effective, both when combined with diet and lifestyle. The most effective non-pharmacological, non-surgical way to lose weight is with very low energy diets. And these are commonly achieved with total meal replacement diets where all normal foods are replaced with formula foods. And so here you have the weight loss compared to all other types of lifestyle interventions, and even 3 years after commencement of a program, greater weight loss has been achieved. What does a total meal replacement look like? Well, we all know what a normal day's food looks like. This is not what I ate on Wednesday, but someone on the internet is what someone else ate on Wednesday. And you can see there's a lot of beautiful regular food. And in a total meal replacement diet, all of that is replaced with this, you know, just some shakes, or sometimes it's in bars. Some new organisations are creating food-based meal replacement diets, but mostly it's formulas. These diets involve severe energy restriction. So approximately 70% of a person's requirements for energy have been cut from their daily energy allowance, and they're only taking in about 30% of what they need. The common dogma is that if a person loses weight quickly with a very low energy diet or a total meal replacement diet, then they will regain the weight quickly. However, that is a myth, because there's now a few studies that have shown that whether a person on average loses weight fast or slow, the rate of regain is the same. Here's one example. It's a study from Katrina Purcell and colleagues in Melbourne in Australia, and they showed that when people lost 15% of their weight with either a food-based gradual diet or a total meal replacement rapid diet, the rate of weight regain was exactly the same over 3 years. But when people lose weight quickly, they tend to lose more weight, and so at the end of 3 years, when they've gained weight back, they're still lower than somebody who lost weight slowly. So rapid weight loss achieved using semi-fasting or very severe energy restriction appears to be the way to go for weight loss, especially when we consider that larger weight losses bring greater health benefits. However, there are concerns about potential adverse effects of total meal replacement diets or the severe energy restriction that they entail on musculoskeletal integrity. And we know that frailty and inability to open jam jars at home on one's own are problems that affect healthy aging. There is also some concerns about potential adverse effects of these total meal replacement diets or severe energy restriction on eating behaviour. So it might be imagined, well, it's in some of the health guidelines that people who do have any tendency towards binge eating disorder and feeling out of control around eating probably shouldn't do total meal replacement diets involving severe energy restriction. But there's very little evidence for this recommendation. So that's why I wanted to investigate full on the effects of these total meal replacement diets and severe energy restriction on body composition, which is the primary outcome, as well as on feeding behaviour. And this was in a project grant supported by the National Health and Medical Research Council of Australia, and it was done in postmenopausal women with obesity. And the reason why I selected to run a study in women and older women is because osteoporosis is 3 times more common in this population than in men. This is some of the wonderful people that I work with. So I am an animal researcher, and I grew up doing experiments with rats and mice themselves, and then started working with humans, and I work with a wonderful team of dietitians, clinical researchers, and psychologists, and all of the team that you need to work with people. So study took women 5 or more years postmenopausal, 45 to 65 years of age. They had a body mass index in the obese range. They had been weight stable for the past 6 months and they were generally sedentary. The intervention involved either a moderate energy restriction for a total of 12 months, so it's a lifestyle intervention, or a severe energy restriction for 4 months, followed by the same moderate energy restriction for the remaining time, up to 12 months. The moderately energy restricted diet consisted of the things that governments around the world tend to recommend that the populations eat. So fruits and vegetables, minimal fun foods or discretionary foods, lean meats, etc. The severely energy restricted diet didn't look very nice. It was 3 to 4 commercial meal replacement products per day, a bit of protein, I'll explain that afterwards, as well as 2 cups of low energy vegetables. Now, the day when I found out that the grant to do this study had been awarded, that was back in 2011, I was at the same time elated and at the same time I was thinking, oh my God, because I promised myself that if I got a grant to do a study on total meal replacement diets in people, then I would do that diet myself for 2 weeks to figure out, so at least I could commiserate with the participants and, not ask them to do something that I wouldn't do myself. But actually it wasn't that bad, because we did some other research into effects of the diet on hunger. And there is actually a hunger suppression that comes from this very severe semi-fasting diet. As I mentioned, both groups, there was protein supplementation in this diet. The reason for that is that protein intake will affect body composition and we wanted to keep this the same in both groups. And physical activity in both groups was encouraged, but it wasn't supervised. Our primary outcome was lean mass after 12 months and also some other parameters of body composition and eating behaviour. Body composition, we went the whole hog and measured it with gold standard techniques. This involved measurement of lean mass with dual energy X-ray absorptiometry or DEXA analysis. We also measured hand grip strength as an index of muscle function because in sedentary individuals, such as the older women in this trial, hand grip strength is a reasonably good indicator of overall upper body strength. We also measured bone mineral density with DEXA of the hips specifically, as well as fat distribution with the MRI scanners. I also made myself go into the MRI scanner as part of my deal with myself for, I wouldn't make anybody go through a protocol that I wouldn't do myself, and that was very scary and claustrophobic, so thanks to our participants. There were 101 women in this trial that underwent randomisation, 50 randomised to the severe intervention, 51 randomised to the moderate intervention. At the end of the 12 months, there were 46 completers in the severe intervention, or 4 women who dropped out, and 39 women completing the moderate intervention, or 12 who dropped out. So already here in the rate of dropout, we had a 3-fold greater incidence of dropout in people on the moderate intervention, or in other words, a 3-fold lower incidence of dropout in people on the fast intervention. And the number one reason for dropout was this dissatisfaction with the diet and with the rate of weight loss. People did not like being, or these 5 people reported not liking being in a trial for weight loss where they were losing, 5 or 6% of their body weight. And I'll get to body weight in a moment. The age of these women was around 58 years, weight was around 91 kilos, and body mass index was in the middle of the obese range, so moderately to severely obese. We just had our data accepted for publication, but it's under embargo, and I'll show you our unpublished results now for all of these, but I'm going to ask you not to tweet any photographs or publicise any photographs from this. First of all, just looking at body weight, so the women in the severely energy restricted intervention, they lost around 18% of their body weight compared to around 6% of body weight at the end of 12 months. We know that severe energy restricted diets result in great weight losses. What we saw also in body weight is that there was a greater proportion of women who lost over 10% of their body weight. Remembering that 10% is what I call the new 5%. Each one of these columns represents one kind person who volunteered to be in this clinical trial. And the length of the bar represents that person's weight loss or weight gain. There are a couple of people who gained weight on the trial. The orange bars are women who underwent the severely energy restricted intervention, and the white bars are women who underwent the moderate energy restriction. And as you can see, there's way more of these orange bars that pass this 10% line than the white bars. In fact, there was 2.5 to 3 times more women who lost 10% or more of their body weight on the severely energy restricted diet compared to women who lost that amount of weight on the moderately energy restricted diet. Now, the primary outcome of lean mass. When we looked at lean mass in absolute terms, we see that both groups lost some lean mass with weight loss over the 12 months. And we know that loss of lean mass, as measured by a DEXA machine, is something that occurs with weight loss. Up to 25-30% of weight loss is lean tissue. What we also saw is that women in the severely energy diet lost significantly more lean mass than women on the moderately energy restricted diet. So they lost about 1.5 times as much lean mass. However, when we normalised the amount of lean tissue relative to the amount of weight loss, we saw that this lean tissue loss was actually commensurate or proportional to body mass loss. So yes, there was greater loss of lean mass, but in proportion to body mass, and both groups lost approximately 22% of their body weight as lean mass. There was no significant difference between the two groups in terms of hand grip strength over the 12 months with the diet. So no loss of strength in either group with the diet. Bone mineral density. There was a problem for bone mineral density. So looking at whole body bone mineral density, there was no significant difference between the groups. But when it comes to hip bone mineral density, with hip bone mineral density being one of the major predictors of osteoporotic fractures in the future, there was actually an approximately 2.5 times greater loss of hip bone mineral density in women on the severely energy restricted diet versus those on the moderately energy restricted diet. So we know that bone mineral density tends to decrease with age. And for women on the moderate restricted diet, the loss of bone mineral density was commensurate with the type of losses that occur over a year in women of that age. But the loss in these women on the severe diet was significantly greater, and that was not accounted for by the greater weight loss. So there's some concern there. In terms of fat mass and distribution, women on the severely energy-restricted diet lost about twice as much whole body fat as women on the moderate diet, and they also lost about twice as much waist circumference. Looking at the abdominal fat in that MRI scanner, we saw that there was also about twice as much loss of abdominal subcutaneous adipose tissue and also abdominal visceral adipose tissue, with abdominal and particularly visceral adipose tissue thought to be one of the most disease-promoting depots of adipose tissue. So we see a bit of a mixed bag of results in terms of the effects of severe energy restriction on body composition in these women. We saw that compared to moderate energy restriction, it did result in about 1.5 times more loss of lean mass, although that was proportional to the extra body weight loss and no difference in hand grip strength. We also saw a 2.5-fold greater loss of total hip bone mineral density, and this wasn't accounted for by greater weight loss. On the outside, there was 2 times more weight loss. In total, about twice as much fat loss, and women were about 2 to 3 times more likely to lose 10% of their body weight and to remain in the trial. I'll come back to body composition later, but before then, for the next couple of minutes, I want to look at the effects of this rapid diet on eating behaviour, specifically on binge eating behaviour. So, as I mentioned, we looked at the effects of both diets on eating behaviour. We did this in 2 ways. One, by a face-to-face interview with the gold standard technique, the eating disorders examination. We also did that with a self-report questionnaire version of that, because people tell different stories when you ask them versus when they tell you by writing it down, as well as loss of control over eating. What we saw is that, contrary to perhaps expectations and hypotheses that the fast diet might increase the level of eating disordered behaviour, we found that in either the face-to-face interview on eating behaviour or in the self-report questionnaire on eating behaviour, we found that women in both groups, well, in brief, there was no difference between either diet in terms of the effect on this outcome, this was a global score on eating behaviour. And if anything, both of the diets seem to reduce behaviours associated with disordered eating. The same was true when we looked at this construct of loss of control over eating. So people with binge eating disorders will often report, and it's been shown in studies, that the feeling of loss of control over eating, regardless of whether a person has objectively or not eaten a large amount of food, is more distressing to the person than the fact that they're eating more food. And we saw no difference between the severe and the moderate energy restricted diet in terms of this effect. So, and if anything, there was a reduction in both diets. So we can see that there's no difference between both diets with respect to the effects on eating disorder behaviours over a period of 12 months. Both diets were associated with either no change or perhaps a slight decrease in these behaviours. So to conclude, with just a summary, we've seen a mix of desirable, neutral and undesirable effects of severe compared to moderate energy restriction. We see mostly desirable effects. We've got twice as much weight loss, and we know that greater weight loss brings greater health benefits. We saw people 2.5 to 3 times more likely to lose that magical number of greater than 10% of their body weight, twice as much fat loss, twice as much abdominal fat loss. There were some neutral effects. If we look at lean mass, where loss of lean mass was commensurate with the amount of weight lost, there was no change in lean function. And in terms of eating behaviour, there was either a neutral effect or potentially a little bit of a positive effect. However, there was this worrying 2.5-fold greater loss of bone mineral density in women on the severely energy-restricted diet compared to women on the moderately energy-restricted diet. What we do not know is whether this relates to a long-term greater increase in the fracture risk, for example, from osteoporosis, and we're currently tracking the 3-year outcomes on bone mineral density. And I think that long-term studies that look at effects of weight management on actual hospitalizations for bone fractures will be important. So to conclude, these results, in light of the fact that obesity is on the rise all around the world, and in light of the fact that there are dose-dependent health benefits of weight loss, in that the more weight a person loses, the more health benefits they seem to gain, I would finish by concluding that clinically supervised, severely energy-restricted diets, for example, achieved in this trial with total meal replacement diet, is a good option for the treatment of obesity, at least in postmenopausal women, but bone health should really be concurrently considered. With that, I'd like to acknowledge all of my collaborators and team, so thank you.
Thank you. I wonder what were some of the age effects for the benefits from weight loss. I mean, the age ranges you mentioned are relatively young, and in older geriatric patients, the results would be completely different. In older geriatric patients, weight loss would be clear sign of frailty. So I wonder how you evaluate the harm versus benefit in those older people.
Older people, yeah. I think that's a really good question, and especially in light of Walter's point about the older organisms and having an adverse effect with older age. I think the only way is to do trials, like to test things. There has been a study published of total meal replacement diets in adults over 65, so I think they were 65 to 85 or so, and there was strength training involved in that, and like supervised strength training, and the effects on lean tissues were, acceptable. But I think really we need to link the data between weight loss and hard health outcomes like hospitalizations and medicine use and things like that. We really don't know.
What was the calcium quantity in both of the diet?
Yes. So both of the groups, according to the dietetics analyses of the diet, were meeting the recommended intake for calcium as well as vitamin D. So the food-based diet was based on Australian guidelines for meeting those recommended daily intakes, and the total meal replacement diet had supplemental calcium and vitamin D included in the product. So they were both meeting the requirements.
Two of your patients there are participants lost 35% of their body weight. Mm-hmm. The only way to do that would be physical activity. Now, you didn't talk about physical activity. That could be also go together with the bone loss. The more physical activity, we know we have damage to bones. So that could be, if you talk, maybe relate to the physical activity.
Yeah. So your point is more physical activity, more damage to bones?
No,
Yeah, right. This is a.
Yeah. Yeah, that's right. So we did measure physical activity. So we gave everybody the same recommendation for physical activity, like, you know, try and be as more active and build up gradually, and it was mentioned in every dietetics group consultation. We didn't supervise their physical activity, but we strapped accelerometers on them for a week at a time, periodically through the year, in fact through the 3 years. And what we found really interestingly was that the women in the severely energy restricted group, they actually increased their physical activity from baseline, including moderate activity and light, as well as moderate to vigorous activity. Whereas the women on the moderate diet, they didn't really increase their physical activity. So there was an increase in physical activity, but even when we took into account that confounder, there were still those significant differences.
Thank you. I suggest we stay seated because there are two questions I would like to approach the panel with. A few words about keto diet and 16-hour diet. What do you think about it? Walter, Amanda, whoever.
Keto diet, most of the epidemiological data is negative. Most of the mouse data is negative. If you look at centenarians around the world, not a single one of the sort of record longevity groups that I know of have a keto diet. Some of the population that have the keto diet, like in northern Canada, they tend to live shorter than the general population. So it's just overall negative.
And about diabetes control?
Yeah, well, for diabetes control, it is short term. We need more studies, but certainly that looks promising. So there could be a use in cancer, there could be a use in some pathology control, but that's going to be tested in clinical trials. But yeah, it seems like if you do it for 2 or 3 months, it probably is not very much of a negative effect. So for example, we're also testing it now in combination with the FMD for cancer treatment. So ketogenic diet plus cancer plus FMD plus standard of care. And then the other one was 16-8, is that what you asked? Yeah. So 16-8, if you look again at the epidemiological data, people that skip breakfast tend to live shorter. They tend to have more cardiovascular disease. So the associations are negative. Centenarians usually don't do 16 hours of fasting. Gallstone formation and gallbladder operation are twofold in people that fast for 16, 18 hours a day compared to those that fast for less than 12. So again though, in treatment of metabolic disorders seems to be lots of positive data. There's a new study that came out from Frank Madeo with 36 hours of fasting that showed a lot of benefits. But Frank, I think, did the right thing also by having lots of warnings and saying, we need more studies on this and actually carefully designed so that it's short term. Another thing that should be mentioned is the metabolic rates. So extreme diets have been shown to, including calorie restriction, to lower metabolic rates, particularly when combined with exercise. And that effect may be epigenetics and it can last potentially years, right? So this is per body weight metabolic effect. So your metabolism will slow down even more than the weight you lost percentage-wise.
And that's a more long stand.
And they can last. This is why lots of people, I think you showed the same thing, regain the weight, because at some point it's even harder than at the beginning to keep the weight. So then you regain it all, and sometimes you regain it more. This was shown a few years ago, I think, with the JAMA New England paper, showing people that exercise a lot and fast, and that long-term fasting, they regain often more weight than their initial weight.
Rabbi, I just wanted to point out there's a Lancet paper I'm looking at right now that shows that when you reduce your total calories from carbohydrates to less than 20%, long-term mortality goes up by 50%.
Yeah.
In fact,
putting yourself in a cage inside lifelong is very dangerous.
Yeah, they show that 80% carbohydrate diet is better than a low carb diet. It's better to go 80% in the Lancet, and there was a meta-analysis, right, Hasi? Better to go 80% than go less than 40.
Thank you, Amanda. That really brings Dear John, again at 2:30, very interesting.
This is probably one of the most important sessions in this conference because we all want therapists eventually at the end of the day. And this session is about how to translate basic research into things that will extend our health and longevity. And some of the leaders in the translation of GEROSCIENCE will present and who will show about the success and failures of this effort, in the hope that we'll all benefit by the results as soon as possible. We start with Professor David Sinclair, who will represent Life Biosciences, a new consortium focusing specifically on geroscience. Of course, he's also from Harvard University, one of the leaders in the field of NAD replacement therapy, one of the few things available that people can use right now to try to extend their health span. And he will speak about the promises and challenges of translating aging research. Please.
Thank you. All right. So I'll give a very short talk. This is going to be 15 minutes long. I'm going to give a bit of an introduction to the field as well of translational medicine. And I know the folks coming after me are going to be fantastic, so I'll get out of the way. You've probably already read this slide. I give this talk fairly regularly. I think most of us are believers in what we do here. But there are a lot of people who are not, who haven't even heard of aging research. And if they've heard of it, they think of crazy scientists injecting themselves with all sorts of things. But there's a truly legitimate group of us, many of us in this room and beyond, who believe that this is going to be the most impactful thing that will happen to humanity in the next 50 years. Making the iPhone look like something that was just a blip in history. Of course, I don't need to remind us, but you can see with the dark blue that there are areas of the world that are increasing in their proportion of older people more rapidly. And these are very large populations, particularly China, Japan in there as well. Europe has increasing burden for health care, as does the US. So we're not just doing this because we like making products and because this is a way to build the economy and make jobs for people, including people in our labs that we train. But we do this, and I do this because I think that this will have a huge impact on the world's economy, saving trillions of dollars each year across the globe. And this is money that can be freed up for even more research, for more training, even helping with other areas of the planetary issues that we have, such as poverty and the environment. And that's a different way to look at this, because if you don't think about it that way, it can seem like a selfish pursuit. And it is not a selfish pursuit at all. It is probably the most altruistic pursuit I can think of. And one of the best solutions to the current problem that we face as a species. What's good, the good news here is that, and myself and many of us have been in this field now for over 30 years, that things have really changed. When I started in the 1990s, even to say that I was going to do a postdoc in aging research resulted in people looking at me funny. Now that's very different. It's great to see the field has come this far. You could argue that there have been a couple of Nobel Prizes, maybe 3 even, eventually awarded in this area, and I think there will be more. And I think that the field does deserve a lot more credit, and it's finally getting there. Now, in science, I think we've got the respect we deserve, but in investment, it's just beginning, and it's a really exciting time. I started one of the first companies in aging research, or aging development, longevity drugs, you might want to call them. This was Certris going back now, what was it, 15 years ago. I can tell you in those days it was tough. Most people didn't believe that this was possible. They certainly didn't understand the concept of addressing multiple diseases at once. And even once you got through that and explained what Geroscience very clearly articulated yesterday, then they said, but how do you sell a drug for aging? And then you have to start giving Neer's talk about aging is one of the major risk factors, and that initially will get on the market, we think, for one disease, but then the drugs will be used broadly. So there's so many things to explain. What we all need to do is to actually be engaging with the public, as many people in this room do regularly, because it's very hard for people to understand the science, the overall global benefit, the business strategy as well. But what I'm seeing, and you can see from this slide, is that finally the message is getting through to a lot of people who have gone from probably skeptics to believers pretty quickly, and it's continuing to grow. So it's a great opportunity. If you have an idea, you have some intellectual property, there may be people out there who can not only advise you, but also fund your ideas. There's never been a better time for that. And while I'm on the topic of advice, many of us in this room have started companies. I think probably everybody here, with very few exceptions, who've been speakers. If you're a young professor or a postdoc and you've got something new, rely on us, come to us. We can tell you all the mistakes we've made, and hopefully you don't have to make them yourselves. We've been learning as we've been going along. You never quit learning, of course, but it's a really tough road, and there's no handbook yet written for how to get this right. And together we can all learn. It's part of the network. So there are a few different biotech models. You can go with venture capital. The benefits are that you get experienced teams, typically, and you get people who will continue to fund you round after round. The venture capital model typically, I can't say for all, but typically is returns within 4 or 5 years, something like that, which is a short time horizon for something like what we do. Alternatively, in the pharmaceutical world, where there's even less, but there is growing interest in longevity research and development, their view is that things need to be really slow and they're totally risk averse. Now, I think there's an opportunity. There are companies you'll hear about today that have a hybrid model where there's a bit more risk because we're doing something that nobody's ever done before, but also the investors and the people involved take a long-term view, and to me that's the best situation. And then perhaps this is the best of both worlds. So I put this slide together based a lot on what I've heard at this conference, and I'm going to touch upon a few of these blue circles here. Now, we've heard a lot about the lower right, and I've picked out the 3 top topics of this conference: drugs that would delete senescent cells, fasting and exercise mimetics, and then we've heard about cellular reprogramming. There are others, of course, but these are some of the hot ones. We'll hear from Natalie in a minute about mitochondrial replacement. That's a really interesting area as well. But I also want to touch upon, in the time that I have, some other areas that I think will lead us into a golden age of health and longevity and push us beyond what Jay was saying yesterday, where we're seemingly maxing out our natural lifespan. We need to get into the world of an unnatural lifespan. We're already there. Anyone who's had an antibiotic is already living an unnatural lifespan, at least an unnatural health span. So the areas are monitoring and artificial intelligence, though I use that word with trepidation. Henry Markram, I think, is in the room. He's the true expert in this. I want to address infectious diseases, in part because I've got a little bit of exposure to that part of the world, but also I think if we don't address infectious diseases, then it'll be a lot harder to extend human lifespan. Most globally, people end up with multiple infections. We've got cell and organ replacement. We haven't talked a lot about that in this conference because this is more about fundamental root causes of aging and addressing that. But I have to acknowledge that that's an area of investment as well and promise. Because if we eventually wear out, we will need replacement, no matter how much we reverse aging. All right, so just briefly, I'll touch upon some things. Now, I didn't show a disclosure slide, I should have. I showed a disclosure slide yesterday. Every one of the companies that I'm involved with, I will mention. And I think without exception, I am involved in the companies I'm mentioning, so assume that I am. This is one that I'm involved with called Liberty Biosecurity. I was a co-founder and I'm an advisor to this company. This is a company that was set up with myself and a friend from the government who knew that there were areas that needed addressing that companies were not addressing, both in healthcare and national security. And this is a company that's working on using biological components of cells, whether they be enzymes or other cell components, to address areas of maximum need in the elderly. And this is one of the biggest ones that's almost never talked about. Anybody who's treated a diabetic will know that one of the biggest problems that they have is lack of circulation, leading, in many cases, to foot ulcers. And this can be very easy. You just rub your foot, you end up with these holes in your foot, these ulcers, which actually have, once you have something like this and the doctors start cutting away at these ulcers, your risk of death is about as bad as a stage 4 breast cancer. And eventually you can lose, and typically you lose a limb. But there's nothing you can really do for this. The antibiotics don't work well. There's biofilms that form across. And so what we've developed with Liberty Biosecurity is a secretion from a fungus that digests biofilms and kills bacteria and viruses. And it seems to be very safe. And clinical trials will begin with this product, which is in this case a powder that will be applied to the wound. The other way of addressing infectious diseases, and I think is going to be a major revolution for the world, including the elderly, is not to just guess what is causing an infection, but actually know what's causing an infection. Right now, if you go into a hospital, the doctor has to guess what it is, right? And then they'll do a test. It can be a bacterial test, grow it on agar plates, 150-year-old technology, or do a PCR test for a virus that you guess is there. Why don't we just say, I don't care what it is, let's just find out what it is, and we can do that with genomic technology. And there are a few companies working on that as well, and this is one that was spun out of my lab and merged with a company from Stanford, run by Carlos Bustamante, founded by Carlos Bustamante. And this is a company that I'm excited about because they're already on the market with a product to detect viruses in organs for transplantation, but the next version of this later in this year, early next, will be a totally disease-agnostic diagnostic. So imagine every kid with a runny nose, every adult that has an infection in a hospital will be tested, and hopefully within a matter of hours we'll know exactly what it is that's there and how to kill it. And right now we guess what antibiotic will be effective initially, and then the test 2 days later will tell you what it might be susceptible to, it probably is. With DNA sequences, we can already predict how to kill these organisms. Monitoring. We all have phones, right? These are monitoring devices. They will become more advanced, they will become more personal. I've seen some of you also wearing a ring that monitors our movement, our heart rate, our sleep. And this will only continue to grow, not only as the technology gets better, but as our kids and their kids will just become so familiar and actually needy of having these monitoring. The idea that you go to a doctor for an annual checkup will seem preposterous to our kids. And this is a company that I didn't start, but I'm a big fan of, called Inside Tracker, which will, for a blood test, you can monitor, I think it's about 34 different parameters, and over time, and I've been doing this for many years now, since 2011, I can see over time what's changing in my body and make corrections. It's the equivalent of having a dashboard on your body, something that I find quite silly right now, that we know more about our cars and how they're functioning than we do about our bodies on a daily basis, and that has to change. And we just published a paper last year in Scientific Reports that used, I think there's about 10,000 customers now, and you can actually mine that data and find really interesting intersections between biochemical parameters And using AI, actually what they do is they give recommendations for types of foods that you might want to eat. And doing that actually lowered people's blood sugar as well as, ostensibly, as well as putting them on metformin. So this is another way that I think we're going to move into the future. It's not only about taking a pill. But speaking of pills, this is one company that I co-founded about 6 years ago called Metro Biotech. It's based in Boston, Massachusetts, and it's an NAD booster company. Their drug in development is a molecule called MIB626, a proprietary crystal form that has been in phase 1 trials for over a year now. And phase 2 will begin in a rare disease or two next year. The thought is to either treat Friedreich's ataxia or another rare disorder like that, which has been shown already in animal studies to be quite effective with these kinds of molecules. And for those of you who are not in this field, NAD boosters work by activating sirtuin enzymes, which you can think of as protectors of the body. And they do other things as well, including improving DNA repair through the proper protein. But it's early days, right? We're still phase 1. We don't have efficacy yet. There's Life Biosciences, which I was encouraged to talk about. I also, before I want to talk about Life, I want to also mention Cobar, which was mentioned by Hassie Cohen. This is also a company that I'm an advisor to, and although I don't have a slide on it, we have some representatives up there in the peanut gallery from the company, and this has been a fantastic company to be involved with because they're actually have been in humans for a couple of years now, and it's really nice to see the advancement of that work. And as you saw from Hassie's talk, just how many peptides are there? There are tens of thousands of these things potentially. We've got so much to mine, and each one of these molecules takes 5 to 10 years to get into the clinic. So this stage of the work is the hardest and the slowest and the most expensive. So we really need to band together and figure out how to make this more efficient. And so one idea that I had with my colleagues and with Nir actually was, how do you bring together our colleagues and our know-how to speed up this process? And Life Biosciences emerged out of that. And this is a company that's headquartered in Boston, but it has offices and labs around the world, bringing together academics and drug developers to try to have an economy of scale, like a pharmaceutical company, but the innovation and the excitement and dynamism of a small biotech. And the idea behind Life was to try and address many of the hallmarks of aging, which you can see here. Now, these are subsidiaries, majority owned and controlled by Life Biosciences, and you can see their names here. I just will highlight a couple of them because I don't have a lot of time. One of them is called Sulfur Gene Therapeutics. It is based on work out of Albert Einstein. This is Anna Maria Corvo's work, and her colleague, Everest. I can't pronounce his last name, but maybe you can. But he's a wonderful chemist. They've figured out what we think is the first way to activate chaperone-mediated autophagy. So when animals are really, really hungry, or a human hasn't eaten for about 3 days, chaperone-mediated autophagy kicks in, and it's a really effective way of getting rid of misfolded old proteins. So that's an exciting company. That company is still in the preclinical stage, meaning that they have many mouse results, and they're hoping by the end of next year to have some efficacy, if not a whole bunch of safety. Okay, next company that I want to highlight is called Continuum Biosciences. So one of the scourges of our age, as we heard earlier, is obesity and diabetes. And this is a company based on molecules that are seemingly safe, but are able to activate mitochondrial uncoupling. So just very briefly, what's mitochondrial uncoupling? Simplistically, it's punching holes in the membrane or making them leaky in the mitochondrial membrane, so cells cannot make as much energy with the same amount of food. Mm. And what that leads to is that your body, or the animal's body, is churning through calories and glucose trying to make energy. And so you get very slightly warmer, imperceptibly warmer, but it's as though your metabolism is running faster. And with animal studies, and there will be some human-like, similar molecules in humans, they do lead to weight loss and improvement in metabolism. And their initial target is fatty liver disease, in an approach that is different than other companies that are approaching this disease, such as Cobar. But ultimately, what I hope for Continuum is that their drug is so safe it can be used by anybody who has a weight problem. And with members of my family who, some of them have weight problems, I can tell you, it's not enough just to say, don't eat, go exercise. That doesn't work for a lot of people. So here's the Life Biosciences Network. We're inspired by Sami. And you can see some of the world-leading scientists that have come together to try and make this possible. I haven't mentioned Steve Horvath yet, who's here, and he's part of this network as well. In a new company that we formed called Iduna. Iduna is the Norse goddess of longevity, and this is the company that is licensing technology related to cellular reprogramming and the epigenetic clock. So that's my overview of some of the things we can do, some of the things that are an opportunity for all of us. And then just to finish up, and I'm happy to take a few questions, I think that what we're really aiming for is the ability to have doctors feel comfortable prescribing these medicines. And I've got to mention that how much will we owe, and do we owe to Nir with his TAME study? Because even though the study is at its early stages, every day people ask me about it, and it's changing people's minds about what doctors should be thinking about when it comes to healthcare. And that alone is a really big achievement, and I wanted to thank Nir for that. Ultimately, what we want is for aging to be a treatable condition, and so that instead of either begging your doctor to give you metformin if you're not diabetic yet, or finding some shady person that'll give it to you, or go to Thailand and get it over the counter, that it will be just a standard of care to be given these molecules that the trade-off is so high in terms of risk versus the great benefit. So I'll leave it there, and I'm happy to take any questions. Thank you.
One or two questions for David?
One here. So some of us in the course of looking at aging research are a bit dismayed that we focus so heavily on mice. But all of can you say whether any of these companies are taking information from other organisms and moving it forward in terms of therapeutics?
So the question was, these companies taking research from other organisms and moving it forward into humans, in the absence of mouse data?
No.
In addition to?
Either in addition or without mouse.
Right. Well, so the answer is yes, but it's rare. Most companies will do mouse efficacy studies in the gold standard models, hopefully multiple models. And some models are terrible, as you know, we all know, Alzheimer's being one tough one. But it's really rare. Sometimes there are studies in dogs. Glaucoma, for example, a disease that we're looking at, can be modeled in dogs. But typically there are no better models than mice. And what higher species, as you call them, are used for are safety studies, and that's it. Okay, so
Can I just add, Hassi gave the example of using genetic data of functional problem, and I gave some examples too to suggest that those are things that are relevant to humans, and pharmaceuticals want to know that. That's exactly the point.
So we could say that going forward, we can still be happy with mice and throw in a few humans from time to time.
Well, you could start with the humans and then go to mice to develop the drug and come back.
I just don't want to cut off all funding for this.
No, please don't do that.
One last question. Yeah.
Just a remark regarding the risks. You were talking about remote sensing and so on, and it's obvious that we are going there, with all the equipment for the elderly especially. But could we really secure that? Could we really be sure that nobody interferes? And just for example, we checked several techniques like blockchain and so on, it doesn't solve it.
Yeah. Right, so the security is the issue, yes? That's the question. If you're giving up your medical data and blood tests and genomes to companies,
Not only if you let somebody in remote sensing correct or help you or remove or adjust your health, whatever.
And the concern is privacy?
The concern is not only privacy, interference or, taking advantage.
Getting hacked?
Cyber
attack. Yeah, yes, I guess we are in Israel. I guess that's something that is always possible, but it's we have to live with risk every day. My car is constantly monitored for speed, even if I'm sitting in the seat, and I give up that data because the rewards are worth it. Well, somebody could hack my car and send it off the road too. Now, it's a personal risk. No one's going to force you to give up your medical records, but I think what is important is, A, that companies like Inside Tracker have extremely good security, and I'm an advisor of that company, and I've been hammering them on that. If you think having your credit card stolen is bad, wait till your blood biochemistry is all over the internet. And also your genome is even worse because you pass that along forever to your offspring. So that's important, and then government regulations outlawing the use of that information against you. And both of those areas have been worked on by industry and government alike.
ילדים שלך גנבו לך את המיטוכונדריות.
Thank you very much, David. Now Natalie Johanna from Minority Therapeutics, a leading company from Israel developing mitochondrial replacement or mitochondrial enrichment therapy. Of course, mitochondria play a crucial role in all age-related disease. Right now they're focusing on the rare disease, but of course this application can be expanded dramatically for all age-related disease. So please, she will talk about mitochondrial activation therapy for rare mitochondrial disease, a gateway for age-related disease.
Thank you very much, Ilya. Thank you, everyone, for this opportunity to present here today. I would just say that sitting here in this audience and hearing all the great ideas that have just started, remember, aging is a very complicated stage, and all of these will have to combine together probably to create a solution, a real solution for this very complex, I don't know how to say disease. I'll say in a moment what kind of a disease. But I think we heard a lot in the past 2 days about different mechanisms that are in the process of aging, such as senescence or telomere shortening, epigenetics effects, and immune system failures. And we haven't touched enough, I believe, in the underlying cause of all of those mechanisms. And I want to take you through a journey through the world of mitochondria. I'm a mitochondrial researcher, so I love to connect everything to mitochondria and tell you about what we are doing in Immunological and how we are studying some of the effects of aging. So we all know that mitochondria is the powerhouse of the cell. That's what everyone says, and it sounds very holistic. It's not Chi. It's a real molecule that is produced by the mitochondria called ATP. And ATP is needed in almost every event that occurs in the cell. For every enzyme that is acting in the cell, it needs ATP to act. So if you don't have enough ATP, obviously, you will not have enough sufficient of the significant pathways that are happening in the cell. But not only that, let's leave aside the ATP production. Mitochondria are also responsible for the building blocks of so many of the events that are happening in the cell. Amino acids, nucleotides, cholesterol metabolism, nitric oxides, glutathione, porphyrins, glucose and fatty acids metabolism, all happening in the mitochondria. We have signaling pathways by ROS or calcium signaling, steroid hormone production, and eventually, if you don't have all of this, the mitochondria will also decide if the cell will die or not through the apoptotic pathway. So it's life and death in the hands of mitochondria. And it was suggested already that aging involves multiple pathways of mitochondrial dysfunction through increased mitochondrial DNA mutations. The mitochondrial DNA is a plasmid. It's exposed completely. It has no protection. And the first DNA that will be harmed in the cell will be the mitochondrial DNA. You have increased ROS generation, enhanced oxidative damage, and of course reduced ATP levels and all other components that I've just mentioned as building blocks. So you have mitochondrial damage, and in a healthy cell, you have a quality control on damaged mitochondria called mitophagy. So if you have a damaged mitochondria out of the mitochondrial network going through fission, the cell's supposed to identify this one as damaged and supposed to remove it from the network. But that's not what's happening in aging and in other diseases. And you get accumulation of damaged mitochondria that are even going through extension. So you get mitochondrial dysfunction that is associated with age-related diseases such as metabolic syndromes that we heard about, neurodegenerative diseases, cardiac diseases, and cancer. My belief is that the best model to study this hypothesis is primary mitochondrial diseases. If you want to look at mitochondria, let's look at the right model to look at to study that. And primary mitochondrial diseases are multisystemic diseases affecting the brain. You see dementia, you see epileptic seizures, strokes, affecting the vision, hearing, cardiac activity, muscle activity, extreme fatigue. You can see liver failure in mitochondrial disease patients. You can see diabetes and kidney insufficiency. Almost every organ in the body is damaged due to dysfunctional mitochondria. The group of primary mitochondrial diseases is considered a rare mitochondrial disease, but altogether they are quite common. They can start from birth to late onset, and as I mentioned, they are multisystemic and there is no available treatment for those diseases. And it was suggested that we all know the 9 hallmarks of aging, that actually the underlying cause of all 9 hallmarks of aging is mitochondrial dysfunction. And I want to touch a little bit on that. For example, this paper showed that in a population that was studied that showed multiple mutations in the mitochondrial DNA was correlated with shortening of the telomeres that eventually creates bone marrow failure and aplastic anemia, which is more common in people in the age of 60 and more. And this is one phenomenon that we can correlate, but of course there are others. But the idea is that we can study mitochondrial diseases and look at age-related parameters such as shortening of telomeres or senescent signaling or immunodeficiencies in primary mitochondrial diseases, and then study aging as a mitochondrial disease and describe events that are associated with mitochondrial dysfunction and determine what is mitochondrial dysfunction during a lifetime of an individual. And that's what we are doing in Minovia while we are developing a therapy. And just to give you a small spoiler, for the same event that we saw here as an aging event of bone marrow failure, we can look at a Pearson patient, which is a mitochondrial disease patient. He has a severe anemia, acellular bone marrow, and cells that are not active enough in terms of colony formation. And when we treat this patient, we can see an increase in cellularity 1 year after the treatment, up to 70% cellularity relative to 20 to 40% before. So we can look at mitochondrial disease patients as with some symptoms that are also common in aging. Some important facts about mitochondria before I describe the therapy itself. They are inherited exclusively from the mother, so we are all carrying mitochondria from our mothers. They contain their own mitochondrial DNA that encodes for 13 proteins only. More than 1,500 are encoded by the nucleus. There are mutations and deletions in the mitochondrial DNA that cause those primary mitochondrial diseases, And there is a phenomenon known as heteroplasmy, where both mutated and white-type mitochondrial DNA reside in the same cell. And that's what will determine the severity of the disease in different organs, the level of heteroplasmy. It was shown before that isolated mitochondria can re-enter cells. And it was also shown that mitochondria can be transferred between cells and rescue the metabolic activity of the cells that receive them. So what we are doing in Immunological is we are developing a therapeutic approach called mitochondrial augmentation therapy, when we are using hematopoietic stem cells as carriers of mitochondria. We are enriching hematopoietic stem cells with mitochondria outside the body, thus changing the heteroplasmy level towards the good mitochondria, and then infusing those hematopoietic stem cells back to the patients. And this is the process. You can see these mitochondria are isolated, frozen, ready for use for the patient. The patient undergoes stem cell collection from peripheral blood. The cells are enriched with mitochondria outside the body, washed and infused back to the patient. And this process, this personalized cell therapy process, takes about 30 hours vein to vein, relatively short process. The first disease that we decided to target is called Pearson syndrome. It's a mitochondrial DNA deletion syndrome that affects only about 100 patients in the world. A very devastating disease that accelerates really fast in those patients. They all die during childhood. There is no available treatment. It starts with severe anemia. They do not grow. They have renal failure, liver failure, diabetes. They have dementia and many other symptoms that we are familiar with. It was shown that allogeneic bone marrow transplantation could work in those patients, and therefore using stem cell therapy could show efficacy. But there are, of course, many adverse events for allogeneic bone marrow transplantation. What we've done is the first in the world a human compassionate use protocol to treat 3 patients with Pearson syndrome. I want to show you 2 examples. I will go on them very briefly. The first one is 30 months after the treatment. It's a single treatment. He had a deletion, a huge deletion in the mitochondrial DNA. 7 genes were missing. He received GCSF for 4 days, we collected CD34 positive cells, and enriched them with maternal-derived mitochondria, since the mother is not a carrier. This is a disease where that is a de novo deletion. This child was 6.5 years old. He was very small. He didn't grow for 3 years. He had kidney insufficiency and another renal Fanconi disease. And he was very, very weak. And more than improving his quality of life, this was amazing, and improving his muscle aerobic function and his gain of weight after 4 years of not gaining weight. Today he's 8 kilograms more. I would like to focus on his kidney as one of the diseases that affects elderly people. So he had a tuberculopathy where he loses all the nutrients through the urine, and he needs to be supplemented through supplements to increase the levels in the blood. That was completely reversed when we are looking at blood magnesium level and blood bicarbonate levels. In the past 30 months, he does not require any supplements, and the urine levels are back to normal. He also had kidney insufficiency with increasing creatinine levels 1 year before the treatment. That's completely blocked, stopped the deterioration of the kidney insufficiency. We've treated 3 patients with Pearson syndrome. Each one of them suffered from a different multisystemic involvement. We observed improvement in all of them in some of the parameters. And you can see, for example, lactic acid, which dropped to normal level in all 3 Pearson patients. And that's, of course, one of the hallmarks of dysfunctional mitochondria. And we also observed an increase in total mitochondrial DNA content in the peripheral blood in all 3 patients. Another one quick example that I would like to give is a patient with Kearns-Sayre syndrome, another mitochondrial DNA deletion syndrome that affects about 7,000 patients in the world. And in this child, who was 14 years old, again, she was very small, not active at all. She couldn't even sit when we were treating her. She had epileptic seizures and stroke-like episodes. She had vision and hearing loss. We could see a dramatic improvement in her brain ECG, as you can see. EEGs are in, as you can see in this figure. The child, 13 months, after the treatment, suffered no epileptic seizures, and her blood ATP levels improved dramatically. And today she can walk with assistance. She sits by herself and walks with assistance. And looking at all those parameters that we've changed in those patients can actually tell us that we could have a systemic effect by manipulating hematopoietic stem cells that's changing so many parameters in their metabolic fate, their ability to produce other blood type cells, to circulate throughout the body, and change metabolic activity also in distal organs. And we have some results. Now we are going back to mice and showing that there are some effect and biodistribution of the mitochondria in distal organs, such as heart and brain, but those are healthy animals. We are going to repeat this in diseased animals to see if there is an improvement. Our next source of mitochondria will be from human placentas. We're going to have a bank of mitochondria, healthy, from the healthiest source, youngest source of mitochondria for human tissues. And we are going to detect the allogeneic mitochondria by using different haplogroups and sequencing methods. We've already been able to show a repopulation of bone marrows of CD34 positive cells that are enriched with mitochondria in NSGS mouse model. And I just want to finish by this. Mitochondria represent an event that occurred during evolution about 1.5 billion years ago. They used to be antibacteria that entered into the cell and enabled the cell to survive in an oxygen-enriched environment. That was the trigger for life, I believe. And as we age, there are diseases that are affecting mitochondrial dysfunction. And we get age-related diseases, all the rare mitochondrial diseases, depending on the dysfunction itself. What Minovia is suggesting is a kind of a re-evolution where we replace or we enrich the level of normal mitochondria in the damaged cells, and specifically in hematopoietic stem cells, which have a way to influence the entire body throughout the circulation. And I want to thank you for your attention and the wonderful people who did the work. It could never have happened without these names. So thank you very much.
Thank you very much, Nathalie. Also, 2 questions to Nathalie, please.
Does the therapy need to be continued for the rest of the patient's life at regular intervals, or is it
So the question is whether we need to repeat the treatment, I guess. So at this point, longest period that we are following is 30 months, and there is no need in repeated treatment so far. We see a continuous improvement in the lives of these children. But again, we need to remember those are primary mitochondrial diseases. We don't know what will happen in the future, but since it's an autologous process, there is no reason not to repeat if needed.
Another question?
Maybe just a comment. This was, first of all, Natalie, I love this work, but I think you have an example of where you went to those poor children, and there are many other diseases that we could then translate to aging, like we hope to do with you. But one of the challenges that David kind of didn't mention, I'll mention it, is when we have things that we're sure is relevant to aging, one of the challenges is to find disease in the meantime that we could start doing, getting money, getting the resources to actually do the aging. So it comes from, both directions.
Exactly. So just to comment on that, Pearson syndrome as an ultra-rare pediatric and deadly disease allows us to come into patients very quickly and to pass a very short regulatory path until approval. We expect to have an approval from the FDA for this therapy in 2 years from now. And then we can have the budget to continue, of course, for the rest of the indications. But accelerating it through collaborations and the passion that there is in this room to move to more common diseases not necessarily for the ultra-rare ones, but rather to the more common diseases as well.
All right, thank you very much. Now it is my pleasure to give the floor to Tal Mir from Tel Aviv University. I think all of us heard about the breathtaking developments from his lab in regenerative medicine. And eventually the real anti-aging therapy will be regenerative therapy to rebuild the structures of aging. And Tal will speak about advanced technologies in regenerative medicine.
Thank you very much, Ilya. I'll just wait until they switch to the computers. Thank you very much, Ilya, and thank you for inviting me to share the technologies that we develop at the lab. So as David mentioned, when we grow older and older, we'll eventually need to replace organs, and this is exactly what we're doing in the lab. We're engineering different types of tissues, including the heart, the brain, spinal cord, intestine, the retina, and others. And I will give a quick overview of the things that we do. Okay, so just to tell you about the concept of tissue engineering, what it is, so we can, in this approach, we can take cells, preferably from the patient, cultivate initially in 2D flasks or petri dishes, and then we need to create tissues. And tissues in the body are not just the cells and the extracellular matrix that actually serves as a glue for the cells, help them to assemble in the proper architecture and eventually to function properly. So we develop in the lab different types of biomaterials. We can add small molecules, nanoparticles, growth factors, or particles that slowly release growth factors. And eventually what we're doing is allowing the cells to assemble into a functioning tissue which can then be taken and transplanted back to the patient. This is a typical cardiac patch that we engineer in the lab. So this is a few centimeters in diameter, a few millimeters in thickness. It contracts nicely in the lab for a long period of time without external stimulation. And if we take these cardiac patches and transplant them on the infarcted heart, for example, we can see a very nice regeneration after 1 month, but if we wait enough, we can see a very nice rejection of these patches, because mostly the biomaterials that are used for tissue engineering are synthetic, and the body doesn't like synthetic materials. So people thought, well, let's take materials from animals, remove all the cells, we're left only with the collagen fibers or perhaps sugars, and eventually this will not provoke an immune response. And we've recently shown that there are certain antigens in pigs, for example, that we do not have in the body, and this will provoke immune response. Additionally, proteins that we do have, ECM proteins like collagens, laminin, fibronectin, they have different amino acid sequence. For example, you can see up to 37% difference in collagen 4, and this will trigger an immune response. So we thought of creating completely autologous patches by relying on the patient's own materials and the patient's own cells. And what we do is we take biopsy, fatty tissue biopsy. We isolate the cells and separate the cells from the extracellular matrix. The cells are reprogrammed to become pluripotent stem cells, and the biomaterial, the extracellular matrix, is processed to become an autologous matrix. Then we can differentiate the cells to whatever cell type that we wish, feed them on the biomaterial, we can create any autologous tissue that we want. And then if we compare the immune response to these materials, comparing between the pig's materials and the human materials and the self-autologous materials, so we can simulate the immune response in a dish. I will not go into the details of this experiment, but I will just say that there is a huge difference between the immune response to pig's materials, even if they're very, very clean from DNA, between pigs and humans, and even between the humans, between individuals, we have differences, meaning that there is a need for personalized biomaterials when we're talking about implants. If we look at this effect in animals, so we can see again that we have less immune response when we're talking about autologous material. So when we transplant these patches, this is how it looks in rats. You can see the patch on the left ventricle, but you can see that there's an open chest surgery, and we thought that it's not ideal for the patient to have an open chest surgery after heart attack. And we decided to create a thermoresponsive hydrogel from these patient-specific materials. So this is how it looks. We process the material to become liquid in room temperature, and now we can mix the cells here, and in 37 degrees it solidifies, it becomes a gel, and then we can inject it wherever we want in the body, to the spinal cord, to the heart, to the brain. These are things that we do at the lab, and this will form the tissue in the body without the need for surgery. So if we think about this platform, so we can take this fatty tissue from the patient, remove the stromal cells, and create induced pluripotent stem cells. The ECM is processed to become a personalized hydrogel. We mix the two together, and now we can create different types of tissues, and this is a partial list of the things that we do in the lab. For example, adipogenic implants for reconstructive surgeries, motor neuron implants for spinal cord injuries, cortical implants for brain trauma, dopaminergic implants for Parkinson, cardiovascular implants. These are things that we do. And we can also use more advanced technologies such as 3D printing and microfluidics to create more complex tissues. So I'll give you some examples. These are the cardiac implants that we create. These used to be fat cells in fatty tissue. Now they're functional cardiac cells. You can see that they contract and they fire. And we've shown that they work in small animals, that they regenerate in infarcted hearts with ejection fraction in small animals. We went to pigs models of myocardial infarction, and we inject them into the left ventricle. We can see nice regeneration. This is preliminary results, but still you can see nice regeneration in terms of ejection fraction. So we can use this biomaterial, this hydrogel, with the cells, patient's own materials to 3D print the patient-specific tissues. So we can look at the CT images of the patient, we look at the blood vessels, isolate them, transfer them to the computer, add more blood vessels to this structure to better nourish the tissue, and then we go to the 3D printer that we have in the lab, and we're printing patient-specific tissues that not only match the immunological profile of the patient, but also the anatomy of the patient. And this is how it looks like. In between these blood vessels, we have cardiomyocytes that contract nicely in the lab for a long period of time. We've already transplanted these patches. We can also 3D print whole hearts. This is something that we've started a couple of years ago. And again, this is from the patient's own materials and the patient's own cells, so it will not provoke an immune response. And you can see the blood vessels are in blue. Of course, this is a very basic heart. The cells contract, but they're not contracting synchronously. But this is the next step. This is what we're doing now in the lab, teaching the cells how to behave, to stick with each other on the electrical level. This is how it looks during printing, and this is the final product. We work also on spinal cord implants, so we, in the same way, we create from the patient spinal cord implant. We implant them, and I'll just show you two movies, but we have many animals. This is the untreated animal that you can see. This is a catwalk, and you can see that one of its legs is pulled, it's not really touching or pushing on this leg. But when we treat these animals, and after 1 week, they already walk nicely. And if we talk about acute spinal cord injury, we have 100% success. If we're talking about chronic injury, we have about 70% success, and this is, I think, really nice. So we can create cortical implants as well. I'm going fast because I don't have a lot of time. Dopaminergic implants that secrete dopamine, and these small implants are already injected to Parkinsonian rats with Danny Offen from Tel Aviv University. We also regenerating the retina, so we create the same implants, and we're just injecting with the ifloboscotol, we're injecting them to the back of the eye. Another thing that we do in the lab is we're engineering cyborg tissues, not just because it's cool and fun to integrate electronics with engineered tissues, because initially we wanted to sense the function of the tissue at all times, to see whether the cells are contracting, if they contract synchronously, if they're dying, and if they do not work properly, we want to be able to stimulate them or release drugs. So if you think about the technology, the patient is sitting in his house, is not feeling well, the physician can log on to the computer, can see the function of the tissue. For example, if he senses that there is not enough beats per minute, can just type on the computer and provide electrical stimulation. If he senses that there is not enough oxygen, can release, for example, VEGF to create new blood vessels. Inflammation, can release anti-inflammatory drugs, dexamethasone. These are things that I'm not imagining. These are things that we actually did in these papers. But sometimes the regular electronics that we're using is not really working well because they're stiffer than the body. And there's organ dynamics. In the case of the heart is moving all the time. There's a mismatch between the mechanical properties, and we thought that we need to create electronics that better matches the body. And we decided to print these electronics, so we formulated new electrodes that can be printed together with the cells. And eventually what we do, we print the patient-specific materials with the cells, with the electronics, together, and now it's really soft electronics that can be manipulated, and eventually we can record the function of the tissue and stimulate the tissue as well. Finally, what we're doing now is creating whole hearts with electronics in electrodes in strategic places. For example, on the septum we have different electrodes to stimulate this area. On the larger blood vessels, we have control release systems that can release VEGF to provide better vascularization, and many other things that we do, and this is how it looks like in CT. With that, I end up, acknowledge lab members, collaborators, my funding, of course, Sami for supporting us establishing the Sabol Center for Regenerative Biotechnology, and thank you for listening.
Thank you very much, Tal. So, two questions once again, please.
I guess it was fast because you have nothing to say really. So the thermal regulation question. So two things. First of all, you're taking on mental fat. How do you do that?
Minimally invasive procedures. 5 minutes
surgery after.
It's a 5-minute procedure that you do a small cut, less than 1 minute.
I wonder why Omentum.
I'll tell you why. Because the Omentum has really great regenerative capabilities. It is used often to you do overlaps on different organs to create the vascularization. So we thought that it would have something to do with the matrix itself. It serves as a depot for stem cells. So we thought that we should use that. So, and it's something that you can spare. You can take it out from the body, and I wouldn't do it for cosmetics, but for really important things like the heart or the spinal cord, the brain. And we take a piece of it.
So I guess the thermal regulation that
thermal
is
response.
there. So if, for example, you want to treat the heart after heart attack, maybe you wanted to go to the core tissues that are the tissues that are not working. Can you use it this way?
Well, it's in 37 degrees it solidifies. If it's cold, it's not it cannot solidify. But the body is in 30.
Not to solidify, to make it liquid again.
So it is liquid. I don't see the reason why to make it liquid again. But the body is in 37 degrees. It doesn't matter where. and then you inject it, and after a few minutes it solidifies. So it gets to the entire area of the scar tissue in the heart, and then it solidifies after a few minutes. And it's not really solidifying; it's becoming a gel with the mechanical properties of the tissue.
Another question? Yes.
Your work is really fantastic. And can you give any example about brain?
About brain, okay, so what we've done with the brain is just in terms of therapy, we've just created the dopaminergic implants, again, taking the same mental tissue, fatty tissue from the patient, reprogramming the cells to become pluripotent stem cells and differentiating them inside the hydrogel to dopaminergic neurons that can secrete dopamine. And we've just started with Danny Offen in Tel Aviv, experiment where we inject these it's small organoids, you can say, droplets of cells that secrete dopamine, so we inject them directly to the...
Do you have real patients?
Just another one?
Sorry? Do you have real patients?
No, not in patients yet. Still in
Just another one for Rafa, please.
Have you looked in the context of aging? Have you taken super old animals that have a completely different immune setup and try to do the implants?
No, but we do take momentum from old people and momentum from young people, and we see differences. And it's you get less stem cells from the. It's something that is known. You see more fat in the older people, so it's different. We prefer to do it with the younger people, but we know that.
Of course, it's clear that part of the technology is going to be applied late in life when you actually replacement parts. So I think that to keep in the context of experimental design and aspect of aging, I think is essential for this technology.
Eventually, we're doing it for the old.
Yeah, of course.
Okay, thank you very much. The last talk for today is from Yael Ben-Venishti, who is the CEO of the Mediterranean Towers venture, the first and so far the only venture fund in Israel focusing on technologies for the aged. Of course, we all want to eventually to create a longevity industry that will generate revenues not just for the companies but for the whole society. And there are many challenges along the way, one of which is to have real evidence-based products, and Yael will speak about creating evidence-based products for the aging population.
Well, thank you. My name is Yael Ben-Venishti. I'm also the chairman of Technologies for Aging Well Group in the Society of Electrical and Electronic Engineering in Israel, and the CEO of Mediterranean Towers Venture, which is the first VC in Israel that invests in technologies for aging well. Unfortunately, we are relatively a small VC, so we are not investing in research, but maybe in the future. So I think that my presentation will be a little bit different from what you've heard in the past 2 days. I will try to stay in the high level and discuss about creating evidence-based products for the aging population. I will start in general to describe the benefits and the barrier in using evidence-based research as a basis for products. Then I will describe a model that will give some solution to these barriers. After that, I will be more focused on the aging research and describe the life course approach. And in the end, I will try to combine everything together and give an example from the area of research on Alzheimer's. So let's start with the benefits. The first benefit that I would like to describe, of course, is that business is that the research on aging contributes directly to the economic savings. Sometimes we can see it on the long run, sometimes we can see it on the short run, but no doubt that there will be an effect on the individuals and on the society. Using health information data analysis, the big data analysis, and using a lot of data that you are collecting in a lot of research, of course, is faster and less expensive than to do experimental studies on each factor by itself and to then search for combinations of factors. That analysis of big data can help us to detect phenomena that are new, that maybe now we don't understand the meaning of them, but in the future maybe this will be the destructive solution for the aging population. And of course, everything is combined together with a trend that's changing our medical world that is called personalized medicine. Now, about the barriers. I think that the most important barrier that we can see is the funding, how we get money into research. So I would like to see the business sector entering the research. But when the business thinking is involved in the research, it can lead to changes in the scientific methods. For example, there can be a delay in publication until a patent is received, or writing a patent on techniques or research methods that will prevent others from using them. And what will happen if the research fails to match the expectation of the funders? There is a threat to the continued funding of the research. And in the worst case, that could lead to false publications. And of course, as in every space, but especially in the space of aging and in medical, there is the barrier of adopting new technologies or clinical protocols. And I think one of the barriers is that the clinicians do not trust the research results. And one of the reasons that they don't trust the results is what I described before, the intervention of the business sector in the research. So the solution that I would like to suggest is actually a new model that takes into consider three components: the academia and the research needs, the business sector needs, and the government needs. The academia should be as free as possible from the intervention of the business sector. They should collect the data that the main role of the research is to collect as much data as possible. For that, there should be a regulation on the time of intervention and the amount of money that the business sector can invest in the research. Also, I think that the academia should hold the IP on the research fundings. That will allow them to get revenues on the short term and not to be so dependent on supports of grants and then on the business sector. Now, the business sector should be market-oriented or product-focused. And they will develop, they will take the IP, of course, from the academia, they will buy it and develop product in the short term. And then they will have the incentive to continue and invest in the long term in order they will get new products that are more advanced. Now, the government role is not only to put regulation on the business sector, but also to be focused and to identify the needs and the challenges in the population, and then they should invest money in the research that matches those goals and in intervention that takes the results of the research and really change the lifestyle of the people. Now I would like to be more focused on aging and present the life course approach in the research. The life course approach offers the opportunity to understand better the connection between biomedical markers and the behavioral, environmental, and socioeconomic factors that are involved in development of age-related disease, called epigenetic. The purpose of life course approach is to identify opportunity and different life stages to promote healthy aging and to reduce the risk of future disability, diseases, and dependence. Now, I will take everything together and try to take an example from the area of dementia and Alzheimer's research. So according to the life course approach, the research is divided into two subjects. The first one is biological, such as understanding the mechanism of neurogenesis or neurogeneration, increasing production of new neurons, we just heard about the mitochondria, but also they are focused on the aspects of environmental and behavioral factors. That means to understand the role of external factors that are related to lifestyle, like learning, exercise, social interaction, calorie restriction, and so on. The business sector are more interested in the environmental and behavioral factors because they can take these results and actually make products out of these, like wearables, IoT devices, all the watches that sensors, and so on. So they can get revenues on the short term. But if we are looking at what's happening outside, we see that there are research centers that the business sector supports. So what are the startups that are in those research centers? We fund the startups that are dealing with early detection of dementia. The question is why? Because there's no cure. So what is the incentive to invest in early detection? The pharma companies are looking for less expensive ways to find the right candidates for their experiments of intervention and new drugs. So they are looking for startups that will help them detect the candidates earlier and in less expensive ways. Now they are sending everybody to do an MRI and to do some clinical testing. They are looking for cheapest way. But they also know that one day there will be a cure, and there will be a cure, then the early detection will be the major component of the treatment. Now the government, shocking, but they are not following the model. There is a national plan for coping with dementia, but they are more focused on reducing stigma and increasing the awareness in the public. They are not investing in research, and they are not investing in intervention that can promote a healthy lifestyle. Now, my last slide I would like to share with you, I think, the fastest way to extend lifespan. Thank you.
So, 2 questions?
It comes with a life insurance policy.
Right, so it's a practical suggestion. So, questions, everybody? Thank you very much. מישהו פה דובר אנגלית במקרה? שואה? לא? כל דוברי אנגלית עזבו, אז אני...
איך הם יענו לך? איך אתה חושב שהם יענו לך אם אתה שואל בעברית?
Does anybody speak English here? I mean,
Everybody speaks English. Ilya, you need to...
So, please sit down next to somebody who speaks Hebrew. Ron is already sitting, and you, Anton, sit next to me or somebody. No, ask you. The lectures, some of them will be in Hebrew, some of them in English.
Google my name.
The speakers want. And all the lectures in Greek? Okay. All the lectures will be in Hebrew, and the one will be in Hebrew, so you will need to translate. Actually, I prepared my in English, but I'll manage. Okay, so I now present to you our presenter, Hen Lieberman, the famous hostess from Channel 10. So now 13.
Great, yeah? So we'll change to Hebrew. Sorry. Okay. If that's okay with you. Wait, how many English speakers do we have here?
Russian.
Okay. How many Hebrew speakers? Democracy. Okay. Okay, we're good. So I'll just say a few words about my connection to this subject. ועכשיו אני אעבור לעברית. אני בכלל כתבת תרבות ביום-יום, אבל לפני כ3 שנים יצא לי לעשות סדרת כתבות על הערכת חיים ושיפור איכות החיים, ואז כך הכרתי את איליה, ואז לראשונה הבנתי את הקונספט של להתייחס לזקנה כתופעה בפני עצמה ולבודד אותה, ואז להתמודד איתה כדי למנוע עוד הרבה מחלות אחרות שנובעות מזקנה. והנושא הזה מאוד עניין אותי, ובגלל זה התבקשתי גם לבוא להנחות את הפאנל. אז קודם, לפני שנתחיל עם כל היושבים כאן, נפתח עם פרופסור ניר ברזילי, שאתה ניר בעצם הראשון שקיבל אישור מה-FDA לערוך ניסוי בחקר הזקנה. אם אני לא טועה, אתה תסביר יותר על זה.
בעברית. בעברית.
לא קיבלת אישור בעברית.
אז בגלל שהפאנל הזה מיועד בעיקר, מה שהממשל האמריקאי רצה כשארגן את הג'ירוסיינס הזה, זה שנתחיל לאסוף את המדענים ולהגיד להם מה התקווה הגדולה שיש בחקר הזקנה, אבל גם לארגן פאנל כדי שפוליטיקאים או אנשים ממשרד הבריאות או עיתונאים יהיו פה ויעזרו לנו להעביר את המסג'. עכשיו, האנשים פה יושבים יומיים ושומעים את זה. אני לא יודע מי פה ממשרד הבריאות והכל, אבל אני מרגיש שאני הולך לדבר אלייך. זה הרצאה אישית. כי אני הולך להציג כמה מהדברים שאמרתי קודם כדי להסביר את זה בשפה אולי יותר פשוטה לקהל הרחב. הנה, נכון? איפה האחרים?
איפה קהל? יש כמה מספיק של משרד
סליחה, וברוכים הבאים, אני רואה פה, צופים שלא ראיתי קודם. בסדר. אז מה שאני רוצה להגיד, אני קודם אגיד עכשיו, אני אראה לכם כמה שקופיות בודדות, גם נתונים ישראלים, על למה אני כנראה צודק במה שאני אומר. מה שאנשים לא יודעים, זה שאפשר לכוון מטרה לזקנה ולעצור אותה. אוקיי, אפשר לעכב אותה, אפשר לעצור אותה, ובממדים מסוימים אפשר גם להפוך אותה, ואנחנו נצליח להפוך אותה בעתיד. אוקיי? כל הדברים האלה אנחנו מצאנו ב-20 השנים האחרונות, וזה חשוב לנו ליישם את זה. זה חשוב לנו ליישם את זה, כי מה שקורה עכשיו זה בלתי אפשרי. אנחנו מקבלים מחלה של הזקנה, כולנו אחרי גיל 60 נקבל את המחלה הראשונה. נקבל את הטיפול למחלה הזאת, המחלה רעה, הטיפול רע, ואז מחלה שנייה, ואז מחלה שלישית, וזה מה שאנחנו עושים בזקנה. אוספים מחלות, אוספים את הטיפולים שלהם, אוספים את תופעות הלוואי של הטיפולים האלה, וזה לא יכול להיות, וזה לא רק שלא יכול להיות, זה לא צריך להיות. ואני אסביר למה. באופן עקרוני, הזקנה, הביולוגיה של הזקנה זה זו שדוחפת את המחלות. עכשיו אתם תסתכלו פה בעולם הזה, ואני אשאל אתכם, למי פה יש כולסטרול גבוה, למי יש פה יתר לחץ דם, אתם לא תצליחו להגיד לנו. אבל אם אני שואל אתכם, מי פה מעל גיל 50 ומי מתחת ל-50, אתם כנראה כן תעשו עבודה טובה. אז זה שלביולוגיה יש זקנה, אתם יודעים, כולנו יודעים. אבל למה אתם חושבים שאם יש ביולוגיה של זקנה, אנחנו לא יכולים לעשות משהו עם זה? אנחנו יכולים לעשות דברים עם כל הביולוגיות. אז זה מה שאני רוצה לספר לכם. אני רוצה לספר לכם איך אנחנו עוברים הלאה, מה עומד בפנינו בלטפל בכל האנשים ולהתחיל לעצור להם את הזקנה. אז הסלייד הראשון שאני רוצה להראות זה פשוט את הקשר בין התמותה ממחלות, ויש פה הרבה מחלות, כתובות באנגלית, לבין הזקנה, אוקיי? וכל המחלות האלה עולות בזקנה. יש עלייה יפה, ותשימו לב, ה-y-axis הוא לוגריתמי, אוקיי? תסתכלו על התקף לב, התקף לב עולה מ-5 ל-5,000, אוקיי? אתם חושבים שכולסטרול זה גורם להתקפות לב? כן, זה גורם פי 3 להתקפות לב, אבל הזקנה זה פי 10. ואני יכול להמשיך, כל המחלות זה אותו דבר, כי מה שמאחד את המחלות זה גורם הסיכון הזה. המחלות האלה הן לא מופיעות בלי הזקנה. גם אם יש לך גנטיקה לאלצהיימר, שתביא אלצהיימר בגיל 60, זה לא קורה בגיל 1, 2, 10, 20, 30, 40. אתה צריך את הזקנה בשביל המחלות האלה, כי לזקנה הזאת יש ביולוגיה. זה לא נכון רק לגבי ארצות הברית, זה נכון גם לגבי הארצה, זה היה מ-2000 וזה מגדי רנט, מקופת חולים. אוקיי, ואתם רואים פה זה לא לוגריתמי, אבל אתם רואים איך זה נוסק מעל גיל 60, איך נוסקים המחלות האלה. עכשיו, מה שאנחנו התקדמנו, ואני אגיד את זה בקצרה במשפט אחד, אנחנו גילינו את המכניזם, את המנגנונים של הזקנה. ואנחנו כבר הסתכלנו, וכשאני אומר אנחנו זה הרבה מדענים, בשיתוף עם משרד ה-National Institute of Aging, שהסגן הדיוויז'ן של ה-Biology of Aging הוא פה איתנו, והם פתחו במבצע כזה, שבו הם לקחו 3 מרכזים ואמרו, אם לך יש רעיון איך לעצור את הזקנה, עם איזו תרופה קיימת, תגיד לנו, ואנחנו נבדוק בעכברים אם זה מעריך את אורך החיים שלהם ואת הבריאות שלהם. ואגב, אנחנו מדברים כרגע על משך הבריאות, אוקיי? איכות החיים היא לא חשובה בינינו. כשאנחנו נתחיל להעריך את הבריאות ויהיה תרופות לזה, אז אנחנו נתנצל שתופעת הלוואי הוא אולי גם אריכות חיים, אבל זה לא מה שאנחנו רוצים לעשות. רוצים להעריך את הבריאות. אז אני אתן פה רק כמה דוגמאות של עכברים שקיבלו את התרופות האלה שכולם בשימוש בבני אדם, והקו הראשון הוא בדרך כלל העכברים של קונטרול לא מקבלים כלום,
ומה שאתם רואים, בואו ניקח את הדוגמה הזאת למשל, זה אלה הקונטרול ואלה שמטופלים. ואתם רואים שהם חיים זמן יותר ארוך, אבל הם גם הרבה יותר בריאים. וזה דוגמאות מאוד אישיות, וזה שאלה אם זה לגברים ונשים. גברים ונשים יש להם מנגנונים קצת שונים, וטיפולים קצת שונים לזקנה, זה דבר אחר. עכשיו, אחד הדברים שאני עשיתי, זה לקחתי משפחה כמו זאת, זה 4 אנשים, זה 4 אחים ואחיות, 2 אחיות ו2 אחים, נולדו בין 2010 ל-2020 בניו יורק. ומה שמיוחד באנשים האלה...
1910.
1910, מה אמרתי?
2010.
כן. עדיין היו חיים ב-2010, וכל אחד מהם הגיע לגיל 102. למעשה זאת האחות הקטנה שמתה קודם, כולם היו בשוק. אוקיי? מה קרה? והאחות הזאת למשל, חיה עד 110, אני סיפרתי אתמול כשאני פגשתי אותה היא ישנה, כבר ישנה 95 שנה, 2 קופסאות ליום. אמרתי לה, אף אחד מהרופאים לא אמרו לך להפסיק לעשן, והיא אמרה, כל 4 הרופאים שאמרו לי להפסיק לעשן, הם מתו. אוקיי? אבל האוכלוסייה הזאת, יש לי 700 מאלה, ואגב, כולם אשכנזים, אוקיי? מסיבות גנטיות, כי זה מחקר גנטי. אבל מה שהאנשים האלה, הם לא עשו שום דבר מיוחד, לא עשו התעמלויות, לא עשו דיאטות, לא אכלו משהו מיוחד, 2% היו צמחונים, לא עשו משהו מיוחד, פשוט יש להם את הגנים לאריכות חיים, ו2 מהגנים האלה כבר הפכו לתרופות בארצות הברית, אחד מרק ואחד על ידי אלון. אבל הסיבה שאני מציג לכם אותה זה משהו לגמרי אחר. השאלה אם האנשים האלה חיו עם מחלות הרבה זמן, שאנחנו לא רוצים או שהם היו בריאים. עכשיו, כשמסתכלים על הבריאות שלהם, אין לי פה את הרשימה, אבל שורה של בעיות של בריאות, אתם רואים שהם חיו 20-30 שנה יותר בריאים. למעשה, אלה בגיל 80, 10% לא היה להם מחלות, ואלה בגיל 130% לא היה להם מחלות. הם מתו יום אחד בשינה שלהם. אבל זה לא מה שמעניין. אוקיי? מבחינתכם. אני אגיד לכם מה צריך לעניין אתכם. לא רק שאנשים האלה היו בריאים, משך התחלואה שלהם היה קצר. Contraction of morbidity, איך להגיד את זה בעברית? הצטמצמות התחלואה. זה משהו? זה ידוע, באמריקה זה ידוע, Contraction of morbidity. אם אנשים חולים 5-8 שנים בסוף החיים, אלה הם 5-8 חודשים בסוף החיים, כמו שאמרתי, הרבה מהם גם לא עושים את זה. ומסתבר שגם ה-CDC מאטלנטה עבר ולקח את הדבר שהכי חשוב, הוא אמר, מה התשלום הבריאותי בשנתיים האחרונות של החיים אצל האנשים שמתים אחרי גיל 100, לעומת אלה שחיים עד גיל 70. והתשובה זה שליש המחיר. אוקיי? שליש המחיר, כשאלה שהם בני 100, כשהם היו בני 70, הם גם לא הלכו לרופא. אוקיי? זאת אומרת, להיות בריאים הרבה זמן, זה חיסכון. לחיסכון הזה אנחנו קוראים longevity dividend, והיה פה ג'רוסקיינסקי שדיבר על זה והסביר איך הוא כתב עבודות שחישב כמה טריליונים של דולרים יחסכו אם אתה תאריך את משך הבריאות אפילו 2-3. שזה אנחנו יכולים לעשות בצורה מאוד טובה. רק להראות לכם, גם גדי רנרט דיבר על חציון, מה זה? חציון עלות שנות החיים. ומה שהוא מראה פה, שבשנה לפני הפטירה, אוקיי, אנשים שמתו ב-70 היו כמעט פי 4 מאלה שמתו אחרי גיל 90. זה נכון בכל העולם, אוקיי? זה הדיבידנדה של הזקנה, וצריך לדעת את זה, כי אנחנו מדברים פה, לא מדברים על טיפול מי יודע מה, על משהו מסובך, על ניתוחים, על כימותרפיה, אנחנו מדברים על דברים פשוטים, שהם לא רק טובים לפציינטים, אלא הם חיסכון גדול למדינה. ואנחנו נשמח לתת לכם חומר על זה, American Federation of Aging Research, אגב, שסטפני לדרמן ועודד ון ויליק יושבות שם, וזה, הם מימנו את המחקרים האלה על ה-longevity dividend. אז רגע, אז מה עוצר אותם? אוקיי? מה עוצר אותם? מה שקורה במחלות אחרות, בוא ניקח כולסטרול, יש לך ביולוגיה, אתה מוצא מכניזם לעצור את הכולסטרול, אתה מפתח את התרופות, התרופה עובדת, ורוצים עוד תרופות כדי לעשות תרופה אחרת, כדי לעשות איזה משהו יותר טוב, וזה מה שקורה ברוב המחלות. הבעיה שה-FDA באמריקה, וגם משרד הבריאות פה, לא מכירים בזקנה כמחלה. Okay? עכשיו, יש לנו בעיה, אנחנו לא רוצים להכיר בזקנה כמחלה, כי יש הרבה, הזקנה היא, זה איזם מאוד גדול כרגע, ואנשים זקנים באמריקה, אנשים לא כל כך זקנים אפילו, אבל אנשים מבוגרים יותר, מפוטרים, הם מקבלים פחות עבודה, פתאום אתה אומר להם גם שהם חולים, ולא כולם חולים, אוקיי? אז, אבל אין איזשהו משהו שכאילו ידחה את הזקנה, או ידחה את המחלות של הזקנה. ואם זה לא קורה, אז אלה שמשלמים על הבריאות לא רוצים לשלם למשהו שאין לו אינדיקציה. ואז גם חברות התרופות לא נכנסות ולא מפתחות תרופות יותר טובות, ותרופות בקומבינציות וכל מיני דברים, כדי שבאמת נתחיל לעשות, להשפיע. תראו, אנשים מתים כרגע בגיל 80, והאפשרות שלנו כבני אדם זה לחיות עד 115 אולי, בטח עד גיל 100. יש לנו 35 שנים שאנחנו לא מנצלים כרגע, ואפשר לנצל מהר. אז אנחנו צריכים לפתוח האינדיקציה הזו, כי בזה אנחנו נטפל בכל המחלות. ומה שאני עושה בארצות הברית זה אני לא חייב לקבל אישור מה-FDA לעשות סטאדי. מה שאני אמרתי ל-FDA, בוא נדבר במה אנחנו מטפלים. כמו שאמרתי, לא אנחנו ולא הם רצינו לקרוא לזקנה מחלה. אז הגענו להסכמה, אנחנו נמנע מחלות של הזקנה. אוקיי? איך לעשות את זה אני לא נכנס. דבר שני, שאלנו אותם, תגידו לנו, זה מה שאנחנו מנסים לעשות, תגידו לנו, אם יש לכם איזה בעיה עם זה, כי אנחנו לא רוצים לעשות מחקר, ואז תגידו לנו, היינו רוצים שתעשו את זה אחרת. אז על זה מה שהזכרנו. את המחקר אנחנו מתחילים בעזרת איפה בסוף השנה או בתחילת שנה הבאה. אז מה שקורה, בחרנו מטפורמין, כי מטפורמין היא תרופה שנותנים לכל החיות בעולם, והן חיות יותר והן חיות בריאות יותר. מטפורמין גם מעכב, לחולים שלא סכרתיים, זה לא מאפשר לסכרת לבוא, לחולים סכרתיים 30% ירידה בהתקפי לב, 30% ירידה בסרטן, 30% ירידה באלצהיימר, ואנשים סכרתיים שמטופלים במטפורמין, חיים יותר ארוכים בצורה מובהקת מאנשים בלי סכרת. אוקיי? זו תרופה חזקה, עובדת יפה מאוד על בני אדם. היה פה מיכאל סלע, אגב, הוא פה עכשיו, בן 95, שאומר, אני 60 שנה על מטפורמין. אוקיי? אז זה אנקדוטה, אבל הוא משייך את זה. כמו שאמרתי, המחקר הזה, זה בעצם, אנחנו משתמשים במטפורמין כמכשיר, בשביל להראות שאנחנו יכולים לעכב את הזקנה, אבל הסיבה שאנחנו בעסק הזה זה באמת לשנות את האינדיקציות, ב-FDA ובכל העולם. אנחנו חושבים שצריך לפתוח האינדיקציות האלה. אנחנו למחקר הזה לוקחים בני 65 עד 80, 3,000 איש, 14 מרכזים בארצות הברית, ואנחנו מנסים להזיז את הזמן עד שמקבלים כל מחלה של הזקנה. כל מחלה של הזקנה. אנחנו לא אכפת לנו, אנחנו אגנוסטים, איזה מחלה יש להם, איזה מחלה הם יקבלו. אנחנו מעכבים את הזקנה, הם לא יקבלו את המחלה הבאה שלהם. אז מה שאמרתי פה זה aging drives diseases. הזקנה מביאה למחלות ואפשר לבלום אותה. עלות הקשישים ביותר, כמו שראיתי, נמוכה מצעירים מהם. בשנת החיים האחרונה זה בכלל סייבינג גדול. תהיה ירידה מבחינה כלכלית בהוצאות אם נשתמש בתרופה שיכולה, אני מצטער אגב, זה איכשהו תורגם קצת אחרת פה במסך שלי, אבל אני חושב שאתם מבינים את המסג' ואנחנו חושבים שאם אפילו בצורה עדינה נשנה את תוחלת הבריאות ב2-3 שנה, אנחנו נחסוך, באמריקה זה $7 טריליון ב-30 השנים האחרונות. אז אני אפסיק פה ואפשר את השאר לעשות בפאנל, או אם למישהו יש שאלה. אם לכם יש שאלה, אם אתם חושבים שלא הצגתי פה משהו שחשוב לכם, אל תיתנו לי לעזוב, או לנו לעזוב בלי שאני אתייחס לזה. תודה.
אוקיי, עכשיו אני רוצה להזמין את יצחק בויק. איפה אתה? אוקיי, בבקשה, יצחק בויק. הוא יושב ראש החברה הישראלית לגרונטולוגיה, שגם אחראי על תוכנית האם הלאומית לזקנה. אז הוא ידבר איתנו על זה. אחרי זה אנחנו עושים את זה לפני. בסוף המצגת של יצחק יעלה איליה, ואחרי זה נתכנס כולנו לפאנל. כאן.
אז תציג כמה דקות.
דבר למיקרופון בבקשה, מר איליה.
יהיו 3 הרצאות מאוד קצרות, כל אחד 3-4 דקות, אני כבר דיבר, עכשיו יצחק, בסוף אני כמה דקות, ואז נתיישב לפאנל, ויהיה פאנל לפי שאלות.
יואב ריבוס. אוקיי, אני רוצה קצת לצאת מהעולם הספציפי הזאת של ביולוגיה ורפואה ונדבר היום על מה שאני קורא בניית תוכנית אב לאומית לזקנה. זה עובד? אני מקווה שכן. עוזר? איזה טוב, איזה פועל? זה בסדר, אוקיי. ויותר ברמה של, הייתי אומר ברמה של מדיניות, בצורה יותר רחבה, כי אני חושב שזה המפתח ליישם כל דבר שנוגע היום לעולם הזקנה. הדבר הראשון שאנחנו חשבנו עליו, עוד האגודה הישראלית לגרונטולוגיה, עוד בשנת 2011, אחר כך ב-2014 פרסמה מסמך מדיניות שאמר שיש צורך בתוכנית אב לאומית לזקנה, משום שהנושא של הזדקנות האוכלוסייה הוא נושא שהוא מקיף בעצם את כל תחומי החיים. את הנושא של הבריאות, של הרווחה, של התעסוקה, של המשפחה, כך שהמערכת הצרכים של האוכלוסייה המבוגרת היא קשורה זו בזו, ולכן גם מערכת השירותים צריכים להיות קשורים זה בזה, ולכן יש צורך באמת בתוכנית אב לאומית שיהיה לה ראייה שהיא מקיפה וכוללנית עם מבט לטווח ארוך. אז אם הדבר הוא כזה רציני, ואנחנו יודעים שהאוכלוסייה המבוגרת הולכת להכפיל את עצמה בשנים הקרובות, ואנחנו רואים שאם זה יעלו הצרכים והיקף הצרכים ועומק הצרכים, אז נשאלת השאלה, האם ממשלת ישראל אכן מודעת לתופעה הזאת של הזדקנות האוכלוסייה ונותנת לה את המקום הראוי לה במערכת סדרי העדיפויות הלאומיות? והתשובה היא כן, ממשלת ישראל אכן התייחסה לנושא הזדקנות האוכלוסייה. החלטת ממשלה מנובמבר 2012 אומרת, בעצם קבעה מספר יעדים אסטרטגיים של מדינת ישראל, ואחד מהיעדים האסטרטגיים היה הזדקנות האוכלוסייה. הממשלה חזרה לנושא הזה ב-2015 וכבר נתנה הגדרה הרבה יותר ספציפית למה המשמעות של האסטרטגית בנושא הזדקנות האוכלוסייה, ואז היא דיברה על הנושא של הגירעון האקטוארי של הביטוח הלאומי, על הבעיה של מערכות הפנסיה, על הנושא של התאמת גיל הפרישה ושילוב מבוגרים בתעסוקה. והחלטה נוספת בסוף 2015 הגיעה לנקודה הספציפית של תעסוקה, ובזה הדבר היחידי בעצם מכל מה שדובר באסטרטגיה הזאת שיושם ויושם בצורה חלקית. עכשיו, גם למשרדי הממשלה השונים, ואנחנו בטח נשמע היום משרד הבריאות מה הוא יאמר, למשרדי הממשלה הספציפיים לכל אחד יש תוכניות. חלקם אפילו מקבלות את הכותרת רפורמה לאומית או תוכנית אב לאומית, וכך יש לנו את התוכניות הלאומיות של משרד הבריאות, וגם התוכנית לאלצהיימר, יש לנו את השלישי בשלייקס של המשרד לשוויון חברתי וכן הלאה וכן הלאה. אבל כל ההחלטות הללו וכל התוכניות הללו למעשה הן ספציפיות, ואין בהן איזושהי ראייה כוללנית שהיא באמת מהווה איזושהי מסגרת ותוכניתה. ואז באמת אני בכלל אומר שכשהולכים לאיזשהו שינוי חברתי צריך שיהיה צ'אמפיון, אז היה צ'אמפיון. וב-2017 חברת הכנסת טלי פלוסקוב הייתה יושבת ראש ועדת המשנה לענייני זקנה של הוועדה לעבודה, רווחה ובריאות בכנסת, ובאותה עת למעשה אני נכנסתי בתור יועץ, נבחרתי בתור יועץ של הוועדה הזאת. בדיאלוג שקיימנו בעצם עלה הנושא שהדבר הכי חשוב לעסוק בו היום זה להכין תוכנית אב לאומית לזקנה. כתוצאה מזה הוקמה הוועדה המיוחדת שאתם קוראים כאן. הוועדה הזאת הייתה מורכבת מנציגי כל המפלגות, והיא החלה את עבודתה. כאשר אנחנו קבענו איזושהי תוכנית שמבוססת על מספר עקרונות. העקרונות שאנחנו העמדנו ביסודה של התוכנית הם קודם כל שכבודו של הזקן, לאפשר באמת את המקום הראוי לו מבחינת מפני שיבה תקום והדרת פני זקן, לשאוף לעצמאות מירבית של האנשים בצמצום התלות שלהם באחרים, לתת לאזרח הוותיק את המקום שלו במרכז, אבל גם להסתכל על אנשים עם צרכים במבט אינדיבידואלי עם מערכת הצרכים האישית, לשים דגש על מניעה, לתת דגש על פעילות הקהילתית של הזדקנות בבית ובקהילה, ולהסתכל על האוכלוסייה המבוגרת יותר כנכס ולא כנטל, ואנחנו יודעים שזה נכס מבחינה זאת שהאוכלוסייה הזאת מגיעה עם הרבה ידע וניסיון, ומחקרים מוכיחים שיש לזה ערך גם חברתי וגם כלכלי. ואז קבענו לנו מהם הנושאים המרכזיים שצריכים להרכיב את תוכנית האב הלאומית. אני לא בכדי שמנו בראש התוכניות הללו את הנושא של אינטגרציה. השירותים בישראל מפוצלים באופן באמת מבלבל גם את האזרח הוותיק ומשפחתו, אבל גם עולה הרבה כסף לשירותים בגלל כפילויות ובעיות במערכות של התיאום והעריכה. והצורך הוא, אנחנו המלצנו על הקמת משרד ממשלתי אחד, שהוא ירכז את המדיניות ואת כל השירותים של נוגעים לזקנים ברמה המקומית, לרכז את הפעילות ביחידה אחת בתוך הרשות המקומית. אני רק אתן 2-3 דוגמאות, לא אעבור על הכל כמובן. כי יש בתוך התוכנית הזאת כמה הצעות מרחיקות לכת, שבהחלט יכולות להיות בוויכוח ובמחלוקת. אנחנו דיברנו על הנושא של מיגור העוני, אמרנו לא יותר תוספת לקצבאות, היום 100,000 שקל, מחר 200 שקל, אלא מדינת ישראל לא צריכה להרשות שיהיו בה אנשים זקנים עניים, ויש למגר את העוני. על זה גם כבר המליצה ועידת מדריד של האו"ם עוד ב-2012, eradication of poverty is one of the major goals, ולכן אנחנו הלכנו על זה. וכן הלאה, אנחנו עסקנו בנושא של הבטחת דיור ציבורי. אתם יודעים שיש אנשים שחיים על קצבת זקנה והשלמת הכנסה, והם לא יכולים להרשות לעצמם לשכור דירה, בוודאי לא לקנות, ולכן יש כ-30,000 איש שממתינים לדיור ציבורי. ואלה הם הנושאים, וכן הלאה עסקנו בכל יתר הנושאים. התוצאה הייתה שאנחנו לא הגענו לסוף הדרך, משום שנפלו עלינו הבחירות ופיזור הכנסת, ולכן מה שנותר לנו זה לקחת את 11 הנושאים אשר אנחנו כן השלמנו את הדיון בהם. הדיונים היו בשיתוף עם משרדי ממשלה, ארגונים ציבוריים, מומחים וכן הלאה, ולצערי לא הגענו גם לג'רוסייאנס להתמודד עם הסוגיה הזאת, כמו שעוד כמה סוגיות שאנחנו לא הספקנו להתמודד, אבל החלטנו שבכל זאת אנחנו ניקח את 11 הדוגמאות הללו, 11 הנושאים הללו, ונסכם אותם בדוח מפורט. הדוח הזה פורסם ביולי השנה, זהו הדוח, ואנחנו החלטנו להגיש אותו ליושב ראש הכנסת. התוכנית המקורית הייתה שהתוכנית תובא לאישור מליאת הכנסת, ואושר על ידי מליאת הכנסת, ומליאת הכנסת תגיש אותו כתוכנית של הכנסת לראש הממשלה, והממשלה תדון בזה ותפעל ליישום. דבר שמחייב כמובן רפורמה והשקעת משאבים. לצערנו הדבר נקטע, ולכן הלכנו ליושב ראש הכנסת, וביולי נפגשנו איתו והגשנו את הדוח עם בקשה מיוחדת שאיננו יודעים מה יהיה, אבל הוא כיושב ראש הכנסת, אנחנו מבקשים ממנו, והוא קיבל על עצמו לקחת את הנושא הזה על מנת שאפשר יהיה להמשיך את המהלך שנעשה, להשלים אותו ולהגיש אותו לממשלה כאן לתוכנן. תודה.
כן, בסדר. אז עכשיו אני אדבר כמה דקות באמת על העבודה שאנחנו עשינו. אז כן, אני לסטמבלר, אני מנהל מדעי של עמותת ותק, התנועה לאיכות ואיכות חיים, שהייתה ברשותו של רפי איתן, זכרונו לברכה, עכשיו אנחנו ממשיכים בלעדיו, ואני גם עכשיו קיבלתי תפקיד של מנהל מחקר ופיתוח בבית החולים גריאטרי שמואל הרופא. ועיקר הפעילות שלי עד עכשיו הייתה לקידום האקטיביזם בנושא של איכות חיים בריאים. זה בעצם הנושא של הפאנל שלנו הוא תוכניות ציבוריות ותמיכה ציבורית במחקר. הקשר הוא הדוק, למעשה השאיפה שלנו להאריך חיים בריאים היא לא רק שאיפה מדעית, היא בעצם שאיפה חברתית. לכן חשוב לתת חלק לחברה, להגיע תמיכה חברתית למחקר כדי לאפשר למחקר לבצע את העבודה שלו. אין פה שום סתירה עם אתיקה מדעית או עם עקרונות מדעיים, פשוט מדובר באפשרות, בתמיכה של החברה במדע כדי שתוכל לבצע את העבודה שלה בצורה אתית ומדעית. שמענו בכנס הזה את ההצדקה העיקרית לג'רוסcience, שעל ידי התערבות תרפויטית בתהליך ההזדקנות אנחנו נוכל לדחות לא רק מחלה אחת, אלא כמה מחלות או מגוון מחלות תלויות גיל, מהסיבה הפשוטה שתהליך ההזדקנות הוא הסיבה העיקרית או הגורם העיקרי, גורם הסיכון העיקרי לכל תחלואת ההזדקנות. אז אם אנחנו נצליח להתערב בתהליך ההזדקנות, נצליח בעצם להאריך תוחלת החיים הבריאים
על ידי כך ניצור רווח כלכלי ואנושי עצום, גם לפרט, גם לחברה כולה. אחד מאבות הרעיון הזה, שבעצם נקרא longevity dividend, דהיינו הרווח שהחברה תרוויח משיפור הבריאות האוכלוסייה הקשישה, היה פרופסור ג'רושנסקי שהיה פה בעולם הזה, אבל עזב כי הוא בעברית, לצערנו. אבל באמת אנשים שבעצם יצרו את התפיסה הזאת, גם ניר שבעצם גרם למהפך בפרדיגמה המחקרית, במקום להסתכל על כל מחלה כישות או כל סימפטום כישות, הוא יצא פשוט לחקור ולבחון ההשפעה של התערבויות על כלל המולטימורבידיות. אפילו בין לנו הגדרה טובה של זקנה או הזדקנות כתהליך ביולוגי, אנחנו נוכל לראות את התוצאות בהשפעה על מגוון מחלות בבת אחת. אז זאת בעצם הגישה שאנחנו מקדמים. אז זאת הצדקה די ברורה, אז כל מי שהיה פה בכנס מבין אותה, באמת דברים די הגיוניים, שכדאי לטפל בבעיה מהשורש יותר מאשר בענפים ובעלים. אז אם הגישה הזאת ברורה, איך אנחנו מקדמים אותה? איך אנחנו גורמים לגישה הזאת של GEROSCIENCE להפוך לעדיפות ציבורית. בקצור, איך אנחנו מביאים מימון ותמיכה רגולטורית לתחום? וכמו בכל נושא חברתי שאנחנו חושבים שזקוק לתמיכה חברתית, אנחנו פשוט הופכים לאקטיביסטים, אנחנו בונים תנועה ציבורית, אנחנו בונים איזושהי שדולה, גם כן אין בזה שום סתירה מעיקרון המדעי, ככה גם חקר הסרטן הפך לעדיפות ראשונה תודות לאקטיביזם ולפעילות ציבורית, ככה גם מחקר איידס הפך לעדיפות ציבורית, ובזכות התמיכה המוגברת אנשים גם, החולים גם שיפרו את איכות החיים והשרידות שלהם, ואנחנו מאמינים שגם חקר ההזדקנות יהפוך לעדיפות לאומית באותה דרך, על ידי הגברה של תמיכה ציבורית, של אקטיביזם. ואז עולה השאלה, האם יש כבר איזשהם תוצאות לפעילות הזאת? במילים אחרות, האם מישהו כבר מעריך חיים בריאים בגלל כל הפעילות האקטיביסטית שלנו? בינתיים לא, אבל עוד מוקדם להגיד. התנועה הזאת לקידום איכות חיים בריאים ומניעת מחלות זקנה, אפשר לומר שהיא תנועה מאוד צעירה, בערך 10-15 שנים, שהיא קצת מתחילה לצות מעל פני השטח. כמובן שהיו תקדימים, אבל כתנועה חברתית עולמית זו תופעה חדשה, אבל כבר יש לה תוצאות משמעותיות, גם בשינוי תפיסה חברתית, גם בשינוי מדיניות. אני הבאתי כאן כמה דוגמאות, שוב התכוננתי באנגלית, אבל אני אשתדל להציג בעברית הקלוקלת שלי, קצת רוסית ויידיש. אז אחת הדוגמאות הקלאסיות, אולי אחד ההישגים המשמעותיים בתנועה האקטיביסטית הזאת היה היוזמה של longevity dividend של ג'רושנסקי, שכבר הזכרתי, שבעצם עשה פלאים בשינוי התפיסה החברתית לטובת אותו הרעיון שעל ידי התערבות בתהליך הזדקנות הנוירוני אנחנו נוכל להגדיל תוחלת חיים בריאים, ועל ידי כך נאפשר רווח עצום גם לחברה וגם כל בן אדם, כי כמובן שלא הכל אנחנו מבטאים בכסף, כמובן שגם רווח נקי בסבל של אנשים חיסכון ברווח, בסבל אנושי ורווח באיכות החיים שלהם. אז זה אולי היה אחד ההישגים המשמעותיים. כמובן GEROSCIENCE Interest Group שה-NIH, בעצם אותה יוזמה שארגנה גם את הכנס הזה, יחד עם עוד מספר כנסים שמתקיימים עכשיו בעולם בשבע מדינות לקידום GEROSCIENCE.
זאת יוזמה שמאחדת נציגים של כל של כל מכוני NIH, כל מי שמאמין בתפיסה הזאת של GEROSCIENCE, שוב שעל ידי התערבות בתהליך הזדקנות נוכל למנוע מחלות זקנה ולהאריך תוחלת חיים בריאים, וזה גם כן היה הישג מאוד משמעותי בהעלאת הפרסטיז' היוקרה המדעית והממסדית של הרעיון הזה. אבל כמו שאפשר לראות, די הרבה מהיוזמות האלה התמקדו בארצות הברית, אבל יש עכשיו גם יוזמות בינלאומיות. אני גאה מאוד להיות שותף לכמה יוזמות בינלאומיות כאלה, למשל מאמר העמדה שניסחנו עם האגודה הבינלאומית לחקר הזדקנות ומחלות הזקנה, שמדבר על הצורך החיוני לקדם חקר הזדקנות ומחלות הזקנה כדי לשפר את אריכות החיים הבריאים לאוכלוסייה הבריאה, תורגם ל-12 שפות, צוטט יותר מ-100 פעמים בספרות האקדמית, הוגש לכמה ממשלות. ברמת האקטיביזם העממי אפשר לומר, אנחנו מארגנים מאז 2013 קמפיין בינלאומי שנקרא יום אריכות החיים או חודש אריכות החיים באוקטובר, כי 1 באוקטובר זה בעצם יום הקשיש הבינלאומי, אז באותה הזדמנות אנחנו גם מעלים מודעות לחקר הזקנה. מאות אירועים התקיימו בעשרות מדינות במסגרת התוכנית הזאת, וזה בעצם באפס תקציב, רק מתוך הרצון של אנשים לתרום ולקדם מודעות לנושא. ותאמינו לו, אבל יש כבר תוצאות לפעילות הזאת. אחת הדוגמאות אולי המובהקות, ב-2017 ארגון הבריאות העולמי פשוט לא הכניס את נושא הזקנה או הזדקנות בריאה לתוכנית העבודה שלו עד 2023, זה פשוט לא היה נושא, ואז הקמנו כל זעקה ציבורית בינלאומית, מאות ארגונים כתבו לארגון הבריאות העולמי ודרשו להכניס את הנושא הזה, וראו איזה פלא, באמת הכניסו פעם ראשונה בהיסטוריה, נכניס את הנושא הזה עם יעדים ספציפיים לשיפור הבריאות של אוכלוסיית הקשישים, והנה אתם רואים את ראש אגף הזקנה של ארגון הבריאות העולמי מדבר על הקמפיין שלנו, אומר שהצלחנו ושה-WHO listened, אז עכשיו זה מופיע בתוכנית, וגם הצהרות כגון הזדקנות בריאה היא אתגר דחוף לכל מדינה, ושתוחלת החיים הבריאים הוא המדד העיקרי ליעילות מערכת הבריאות, זה גם כן הופיע בזכות הקמפיין הזה. עוד הישג של אחד הארגונים שאין שייך להם, של International Longevity Alliance, יחד עם Gerontology Research Foundation, זה שהצליחו להכניס את ההזדקנות כאחד מ-modifiers, כאחד מגורמים בהגדרת מחלות במסגרת ה-ICD, International Classification of Diseases. זאת אומרת, הזקנה עוד לא מוכרת שם כמחלה, אלא כגורם סיכון, אז זה מה שהמודיפייר הזה עושה, וזה בעצם יכול לעודד תעשיות וגופים מחקרים להשקיע בתחום הזה. עדיין ההגדרה הזאת לא מספיקה, צריך גם מדדים, צריך לפתח מדדים שיהיו בקונסנזוס, כדי שנוכל באמת לאבחן את המצב הרפואי הזה, אבל כבר יש איזושהי התקדמות וכמובן דברים משתנים. אז רואים שאפשר להגיע להישגים גם בשינוי מדיניות ברמה עולמית, אבל האתגר האמיתי הוא ברמה הלאומית, כי רוב המחקרים והפיתוחים מתבצעים כאן, וגם רוב המימון ניתן ברמה הלאומית, ולכן חשוב לשפר את המדיניות ברמה הלאומית, וגם כאן היו כמה הישגים. קודם כל הצלחנו באמת להכניס באיזושהי צורה את הנושא של הגברת מחקר, פיתוח וחינוך לקידום אריכות חיים בריאים ומניעת מחלות זקנה לתוכנית אב הלאומית. כמו שיצחק אמר, זה עדיין דוח ועדיין בתהליכי דיון, אבל לפחות שמנו שם איזושהי דריסת רגל לנושא, עוד לא כתוכנית פעולה, אלא כנושא אסטרטגי לדיון. וזה תודות, הייתי אומר, לשיתוף פעולה, לברית אחים בין העמותה שלנו, עמותת ותק, בהנהלת רפי איתן, ועמותת נכה לא חצי בן אדם, בראשות אליס פרידמן וחנן טייל, שגם יושב פה. ארגנו כמה דיונים בכנסת, כמה ישיבות ועדת הכנסת בנושא הזה של קידום מחקר ופיתוח, והיו גם כמה תוצאות מעשיות. אני לא מתבייש להגיד אפילו מול הגורמים הממשלתיים שאפשר לומר בתרומה מסוימת של העלאת המודעות בנושא. גם יצאו כמה קולות קוראים בתחום, למשל קול הקורא של משרד המדע בנושא הזדקנות בריאה עם דגש על גנטיקה ואפיגנטיקה. עכשיו גם בקרוב יצא קול קורא של שיתוף פעולה דומה בין ארצות הברית לישראל בתחום הזקנה, וזה תודות ליוזמה שלנו יחד עם הארגון האמריקאי שנקרא Global Health and Policy Institute בהנהלת דינה רוג'רס, שגם הייתה פה לפני כמה זמן ועזבה. אז כאילו זה כבר נכלל כחלק מחוק הסיוע של ארצות הברית לישראל. את יודעת, 4 מיליארדים לסיוע צבאי ו2 מיליארד דולר לתחום הזה, אבל כבר איזושהי דריסת רגל שאפשר לפתח, ואני מאמין שאנחנו נמשיך, וגם בכנסת הבאה אני מאמין שאנחנו נמשיך בקידום הנושא, גם כדי להגביר את התמיכה בתחום בישראל וגם כדי להגביר שיתופי הפעולה של ישראל עם שאר המדינות, כדי שהנושא הזה יצמח ויצא פרי, לא רק כאן אלא בכל העולם, כי הדבר פשוט בנפשנו, החיים שלנו, החיים של הקרובים שלנו, הבריאות שלנו, תלוי בהצלחת התחום הזה, ויפה שעה אחת טוב. תודה רבה.
אוקיי, אז אחרי ששמענו את הצדדים המחקריים, חברתיים, אקטיביסטים, עכשיו אנחנו נרצה לדעת מה במשרדי הממשלה גם עושים בעניין הזקנה, מה הפיתוחים, כמה תקציבים מופנים לעניין הזה, אז עוד מעט, כן, אז בואו נראה.
אגב, הפאנל גם מוחלט, אז מי שרוצה שיפיד אותו, שילחץ פה במשהו.
אז נמצאות איתנו דוקטור שרון יגור קרול, מייצגת כאן את משרד המדע והטכנולוגיה מתחום מדעי הרפואה, ביחידת המדען הראשי. אם יש עוד תפקיד שאת רוצה להוסיף, אוקיי. לצידה יושבת דוקטור איריס רזולי, מנהלת המחלקה לשירותים קהילתיים באגף לגריאטריה במשרד הבריאות. את איליה כבר הכרתם, וגם את יצחק בריק, ושלום גם לדוקטור חגית שווימר, מנהלת מחלקת מדעי החיים במנהלת הישראלית למחקר ופיתוח אירופי, שזה בעצם גם ייצוג של משרד הכלכלה. הרשות החדשנות. בסדר גמור, ויש לנו כמובן גם את פרופסור ניר ברזילי ששמענו, ואנחנו נחזור גם לשלושתכם עוד מעט עם שאלות, אבל אני רוצה להתחיל איתכם. קודם כל נתחיל איתך שרון. את יכולה לספר לנו על התוכניות שיש במשרד המדע והטכנולוגיה שעוסקות בזקנה, וגם כמה תקציבים מופנים לעניין הזה?
כן, אז קודם כל משרד המדע שומעים? אז משרד המדע כבר ב-2002 היה שותף להקמתו של מרכז ידע להזדקנות, שכרגע עדיין קיים ופועל, ואנחנו כבר לא שותפים לו. ובשנת 2015 פורסם קול קורא לראשונה בתחום ההזדקנות, שקול קורא הזה קוראים מדע, טכנולוגיה וחדשנות למען אוכלוסייה בגיל השלישי. זה הקול קורא מיועד לגיל השלישי, כשמשרד המדע בעצם, המחקרים שלנו אמורים להיות מחקרים פוטנציאליים בין המחקר הבסיסי, להעביר אותם אחרי זה למחקרים תעשייתיים, יישומיים. אז בקול קורא הזה הוא פורסם 3 פעמים, פעם ראשונה 2015, שנייה 2016 ושלישית 2018. סך הכל לכל שלושת הקולות קוראים הוצב כ-40 מיליון שקל, ומונו במסגרת שלו 43 מחקרים ספציפיים בתחום של הגיל השלישי. יש לי פה סתם דוגמאות לסגנון של מחקרים, למשל מחקרים היו מאוד מגוונים,
טיפול לניוון מולקולרי של גיל שלישי, רובוט שירות, התערבות תזונתית לגיל השלישי, כאלה ואחרים מאוד מגוונים ששילבו ידע ברפואה, טכנולוגיה וחברה. עכשיו גם ב-2020 עכשיו פרסמנו קול קורא חדש, זה קול קורא שהיא הזכיר אותו, שזה הזדקנות בריאה, גנטיקה ואפיגנטיקה, ששם המטרה זה באמת להביא לידי יישום את ההבנה המולקולרית איך תהליכי זקנה קורים, המחלות שקשורים לזה. לכל קורא הזה הוא קצת כרגע 9 מיליון שקלים, וזה נמצא בתהליך של שיפוט.
עכשיו אם את מסתכלת על האיזון בין פיתוחים טכנולוגיים שעוזרים לאיכות החיים של האוכלוסייה המזדקנת, לבין פיתוחים טכנולוגיים שיכולים באמת לבודד את הזקנה כתהליך בפני עצמו ולמנוע זקנה או למגר זקנה, איפה את רואה את החלוקה הזאת?
אז אנחנו לא כיוונו למשהו ספציפי, באמת החלוקה היא מקרית, זה בערך 50%, וגם חשוב לציין שבקולות קוראים אחרים זה לא מכוון לאוכלוסייה של הגיל השלישי, אבל כולם יכולים להגיש, ושם גם יש נושאים שרלוונטיים כמו רפואה רגנרטיבית, שרלוונטי פתוגנים עמידים, ואם בן אדם מגיש בהקשר של גיל שלישי, זה כמובן משפט בפני עצמו בלי שום קשר. אז אין איזה משהו, העדפה לתחום זה או אחר. אנחנו גם בקטע שמממנים מחקרים של הנדסה ופיתוחים, וגם ביולוגים עם פוטנציאל יישומי, לא מחקר בסיסי.
אוקיי, תודה רבה, ונעבור לאיריס רזולי. כיצד?
דוקטור איריס רזולי מוסרת התנצלות.
אוקיי.
היא חולה, אני דוקטור שלי שטרנברג, אני עובדת איתה באגף לגריאטריה במשרד הבריאות.
מצוין. אז אם תוכלי להסביר לנו איך מערכת הבריאות נערכת להזדקנות האוכלוסייה, הערכת תוחלת חיים, אבל לא הערכת תוחלת חיים בריאים עדיין.
אוקיי, אז ההיערכות מתבססת על 3 מטרות על זקנה בריאה, אז healthy life expectancy, פחות צורכי בריאות, כגון לשים דגש על פעילות גופנית, ופה יש צורך מאוד משמעותי במחקר איך גורמים לאנשים בכל הגילאים, ובמיוחד בגיל המבוגר, לשנות אורחות חיים. העיקרון השני הוא מודלים חדשניים מקצועיים המתאימים לאוכלוסיית הזקנים ושירותים המותאמים לזה של האוכלוסייה הזקנה. יש הרבה בעיות מורכבות במערכת שלנו, שמה דגש על טיפול במחלה אחת. כמה דוגמאות שאנחנו שמים דגש עליהם זה להעביר את כל הטיפול הביתה. יש לנו גם תוכניות לאומיות שהוזכרו פה, שמבוססות על הסינדרומים הגריאטריים. יש 5 סינדרומים גריאטריים עיקריים שהם אי שליטה בשתן, נפילות, ירידה קוגניטיבית, סוף החיים, שינויים נורמליים עם הגיל, שימוש בתרופות, ויש לנו 3 תוכניות לאומיות, 1 על דמנציה, 1 על מניעת נפילות ו1 על טיפול פליאטיבי בסוף החיים, ובשנה הבאה אנחנו נפתח תוכנית לאומית גם בנושא שימוש מושכל בתרופות. העיקרון השלישי הוא באמת לוודא כמות מספקת של שירותים ביחס לאוכלוסייה, כגון כוח אדם, מיטות אשפוז, מספיק צנתורים וכו' וכו' וכו'. אז מבחינת מחקרים, בינתיים רוב הדגש היה במחקרים פנימיים במסגרת התוכניות הלאומיות האלו, אבל בהיערכות יש פרק שלם על מחקרים, ואלון נמצא פה, ממשרד של המדען הראשי, שהמחקרים יחצו את כל הנושאים האלו. אז נגיד בתוכנית הלאומית הפליאטיבית מומנו 3 מחקרים יישומיים קטנים, אחד מהם לדוגמה בדק את היעילות של תוכנית להכשיר רופאי משפחה בנושאים פליאטיביים. בתוכנית לאומית לדמנציה, להתמודדות עם מחלת אלצהיימר ודמנציות אחרות, יש לנו מחקר הערכה על כל תוכנית שאנחנו עושים. לדוגמה, מימנו תוכנית התערבות בכל הקופות, והמחקר הראה שכן העלינו מודעות וידע באנשי מקצוע. אנחנו גם מריצים עכשיו תוכנית HELP בכמה בתי חולים גדולים בארץ. התוכנית הזאת במיוחד ברמב"ם ואיכילוב, זה באמת שינוי תפיסה איך לטפל בקשיש בבית חולים. מסתכלים על הבן אדם באופן הוליסטי ומשתמשים במתנדבים להתמקד בהולכה, שינה ומניעת בלבול שנייה, את המרכיב השלישי, אוקיי, לא משנה. אז זה עשה שינוי בתפיסה איך לתת שירות לזקן בבתי חולים, אז יש לנו מחקר בנושא הזה. עשינו סקר ארצי עם מכון לבקרה על מחלות קרוניות, דוקטור ענבר צוקר, ואנחנו יודעים עכשיו שיש כ-100,000 אנשים במדינה החיים עם דמנציה, ואנחנו יוצאים עכשיו עם סקר חוזר. זה היה הישג, כי כל קופות חולים שיתפו פעולה. יש לנו תוכנית של ידידי דמנציה, דמנציה פרנדס, לגייס צבא של מתנדבים, ויש מחקר מלווה גם כן. אנחנו שותפים במחקר שייר, הממומן על ידי אזרחים ותיקים. אנחנו גם בשיתוף פעולה עם מחלקת המחשוב והחדשנות במשרד הבריאות. יצאנו כמה מכרזי אתגר לפתח טכנולוגיה למניעת נפילות ולטפל בחולה הסיעודי. אז רק לסיים, אם נשים דגש על כמה נושאים ספציפיים, אז נתמקד בתוכנית במנגנוני, ועוד פעם, הכל עדיין בדיונים, אבל יש מודל שכבר אושר. במנגנוני הקצאה נדון בקאפ החדש, בקפיטציה, בכשל שוק, בגריאטריה פעילה, במיטות לסיעוד מורכב. מבחינת כוח אדם, אנחנו נבדוק במחקרים איך למתג את הגריאטריה שיהיה יותר אטרקטיבי לעובדים. אנחנו נגדיר מטפל סיעודי כעיסוק בפני עצמו שהיום לא קיים. יש הרבה דגש על הטיפול בקהילה ובבית. יש במסגרת הרפורמה בסיעוד 11 תוכניות של תמריצים כספיים לשפר את הטיפול של הקשיש בבית, כולל שיקום בבית, כולל הוספיס בבית, כולל טיפול הוליסטי לאנשים עם בעיות מורכבות. ומבחינת הזדקנות בריאה אנחנו נשים דגש לא רק על מערכת הבריאות וקופת החולים, ששם נתמקד בחיסונים, נגדיר ביקור בריא, אבל גם בטכנולוגיה, ברשויות המקומיות. אנחנו רוצים לעודד עיר ידידותית גיל, וגם עם משרדו של ראש הממשלה לשים דגש על עיסוק ותעסוקה. כמובן לא שמתי דגש, אבל כל הנושא של לטפל במטפל העיקרי בבני משפחה גם תופס תאוצה מאוד מאוד משמעותית בתוכנית.
אם אני צריכה לדרג את כל הקשיים שעומדים היום בפני מערכת הבריאות, איפה היית ממצבת, איפה היית מדרגת את גדילת האוכלוסייה הזקנה בישראל? כמה נטל וכמה קושי זה מוסיף להתמודדויות של מערכת הבריאות היום?
אני גריאטרית, אז I'm biased, אז אני שמה את זה כאתגר הכי משמעותי היום לפני מערכת הבריאות.
כן, את יודעת לומר לנו כמה, יש מחקרים שונים שיוצאים בנושא הזה, אבל כמה אנשים מעל גיל 60 חיים כיום בישראל מול כמה ילדים מתחת לגיל 5? יש חלוקה מאוד אלארמינג, הייתי אומרת.
להגיד, התברכנו להגיע למיליון קשישים. השנה אני לא יודעת בדיוק כמה גרים מתחת לגיל 5, אבל יש נתון כזה של האנשים בגיל העבודה לבין הקשישים, כן, הערכת התלות, והמספר הזה עולה ומאוד מדאיג. יש לי כמה נתונים פה. בישראל יש פער של 10 שנים בין תוחלת חיים לתוחלת שנות החיים הבריאותיים, אז חייבים לגשר על הפער הזה. בני 65 פלוס מהווים 20%, יותר מ-20% מסך הביקורים במרפאה בקהילה. פה אומרים ממוצע של 8 פניות בשנה לרופא, אני חושבת שזה אפילו יותר. זה מאוד מדאיג, כמעט 20% מנשים ו10% מהגברים מדווחים על תחושת בדידות, ולא מדבר על הגידול בהוצאות בקשישים, גם בתרופות, גם בשירותים קהילתיים, גם בשירותים באשפוז וכו'.
ועדיין, לפי התוכניות שמנית, נראה שהגישה במשרד הבריאות לפחות, היא עדיין טיפול באיכות החיים של אותה אוכלוסייה מזדקנת, עדיין לא מדברים כל כך על מניעה, על מיגור זקנה. זה לא נמצא שם כל כך.
נכון. בשלב הזה לא מדברים עדיין על מיגור זקנה, אני מניחה ככל שהמחקרים של פרופסור ברזילי יצאו בפועל, אז יהיה יותר מיקוד בנושאים האלו, במיוחד אם יהיה טיפול.
כן, בסדר, תודה. עכשיו נעבור אליי דוקטור חגית שווימר. בואי תספרי לנו אילו טכנולוגיות מסייעות מול טכנולוגיות מונעות ועל שיתופי פעולה גם בינלאומיים.
אז אני פה, כמו שאמרת, מייצגת את המנהלת שיתוף פעולה ישראל-אירופה, יותר מאשר שאני מייצגת את הרשות לחדשנות. ובתוקף זה את התוכנית הורייזן 2020 לקידום מדע וחדשנות של הנציבות האירופית. אירופה מקדימה אותנו בכל העניין הזה, היא מקדימה אותנו בגלל שהבעיה הרבה יותר חמורה, הרבה יותר אחוז המבוגרים באוכלוסייה הרבה יותר גדול מאשר בישראל, ומבחינתנו זה טוב, כי כשהם מתכוננים אז אנחנו יכולים ללמוד.
בעצם אצלם זה קורה דווקא בגלל דברים חיוביים, כמו הערכת תוחלת חיים.
עלייה באחוז הזקנה זה תמיד חיובי, זה אומר שזה גיל מבוגר וזה מצוין, שם
אבל יש לזה תופעות לוואי בעייתיות.
באירופה באופן ספציפי, אחת מתופעות הלוואי שיש גם פחות אוכלוסייה צעירה, ואין מי שיתמוך באוכלוסייה המבוגרת.
באחוז ילודה נמוך.
נכון, בדיוק. בדיוק, ולכן הפיזור באוכלוסייה, השינוי הדמוגרפי, כמו שהם קוראים לו, זו הבעיה המשמעותית, ובאמת בישראל הבעיה הזאת, אני חושבת, פחות משמעותית, אבל אנחנו לומדים ממי שזה חמור אצלו. אז במסגרת התוכנית הזאת, בשיתוף פעולה ישראל-אירופה, התוכנית המסגרת הורייזן 2020, שבוודאי חלק מהקהל מכיר בתור התמיכה כ-ERC במחקרים, אבל חלק משמעותי מהתוכנית הזאת עם כמות מאוד משמעותית מהתקציב הולך למה שהם מכנים אתגרים חברתיים בכל תחומי המדע באופן כללי, והזקנה היא חלק משמעותי כנושא בינתחומי, גם בריאות, אבל גם כל מה ששמענו עד עכשיו, סוציולוגיה ופסיכולוגיה, ופיתוח טכנולוגיות שיוכלו להקל על הזקנה, שימוש בכלים דיגיטליים ו-ICT וכל הכלים החדשניים האלה, ובאמת התייחסות גם להמון נושאים מעבר לבריאות, כמו שיתוף בקהילה ומניעת מידור חברתי ושמירה על רווחה באופן כללי. אז קודם כל פה אני באה להגיד מצד מדינת ישראל. מדינת ישראל כבר נתנה את כל הכסף. הכסף נמצא באירופה ומחכה לחזור לישראלים. והישראלים, ישראל היא מדינת אסוציאציה לאירופה ובתוקף זה זכאית לכל התקציבים. וישראלים כולם, רופאים וחוקרים באקדמיה ובתי חולים ומשרדי ממשלה ועמותות וכל מה שיש לו איזושהי נגיעה לעניין, יכול לקחת חלק בתוכנית ולבדוק אם יש קול קורא לפרויקט ספציפי ולהיכנס ממש לתוך אירופה, לשיתוף פעולה עם אירופה, עם אלה שזה מאוד מאוד בוער אצלם וללמוד מהם ולקדם אצלם את הדברים.
אוקיי, ומוטיב חוזר בשאלות שלי, זה באמת שוב כמה הגישה היא יותר לטיפול בבעיה וכמה היא יותר מוכוונת מניעת הבעיה.
הטיפול בבעיה, גם כשמתייחסים לבריאות פה, אז אם יש התייחסות לטיפול באוכלוסייה המבוגרת, ולשים את הקשיש במרכז, והתייחסות לקהילה, ואיך שהיא מתייחסת. זה לא מיגור, זה טיפול בבעיה, זה הקלת הסימפטומים אולי, אבל גם טיפול בריאותי שהוא לא חלוטין טיפול בבעיה.
אוקיי, תודה. אני רוצה לעבור אליך, פרופסור ברזילי. כשאתה מדבר על חקר הזקנה, על ניסויים בחקר הזקנה, ראיתי שיש עניין אם לא לרצות לקרוא לזקנה מחלה. כמה באמת הרצון לא לפגוע גורם לנו לא להתייחס לזה כבעיה, ואז גם לא לחקור את זה כבעיה.
לא, אני חושב שזה מילים. אני רק רוצה להגיד משהו, אני חושב שפה הייתה הדגמה נהדרת של הבעיה. כל הכבוד למה שאתם כולכם עושים, ולא צריך להפסיק את זה. אבל אתם מתכוננים לעתיד יותר גרוע ולתת יותר שירותים. ולהגיד, אנחנו עוד לא עושים עד שלא יהיה המחקר, זה לא מקובל עליי. תשמעו, היו פה אנשים, גם האנשים מאמריקה וגם האנשים מישראל, הם היו באותה רמה. יש פה ביוטק בנושא של הזקנה. יש פה אנשים שיכולים לעשות את ישראל מספר אחד. ואני לא יודע למה אתם חושבים שאתם צריכים לחכות. זה לא מקובל עליי. ישראל צריכה להיות שנייה. אוקיי? אני לא יודע מה, אנחנו באים ואומרים לכם, אפשר לטפל בזקנה, אתם אומרים, אנחנו לא מוכנים לזה. או שהכספים באירופה. אנגליה לפניכם, סין לפניכם, סינגפור לפניכם, כל מקום בעולם חושב על זה, מדבר על זה, מזמינים מדענים. אני ממש שמור, אני לא מבין, זה המדינה הכי מתקדמת, עם האנשים הכי מוכשרים פה. למה אתם אומרים, אנחנו לא מדברים על זה? כל העולם מדבר על זה. מה, פה אין בעיה של זקנה? יש אותה בעיה של זקנה. אז יש יותר ילודה, אבל מה, הזקנים גם כן מזדקנים.
אפשר הערה של זקן מהיציע של החבובות?
כן.
הזקן במרכז הכתם העיוור.
הזקן במרכז הכתם העיוור.
כן, זה בשבילך.
תודה רבה.
שם הכתבה.
אבל זה בדיוק השאלה ששאלתי אותך מקודם, ועד כמה יש פה בעיה של גישה, בעיה של תפיסה שצריך לשנות. אולי אתה תוכל לענות על זה, כלומר, כמה קשה לשכנע שזקנה זה בכלל משהו שאפשר לפתור. אני חושבת שרובנו רואים את זה פשוט כמשהו שהוא חלק בלתי נפרד ובלתי נמנע מהחיים. ולכן בכלל השימוש במילים מיגור הזקנה לא מסתדר לאנשים כל כך. לא נשמע כבעיה הגיונית.
רוצים את הירושה.
מה שאנחנו מציעים, מה שאנחנו מתקדמים, גישת הג'רוסנס היא גישה מאוד הגיונית. אולי הגישה ההגיונית היחידה שמאפשרת לנו להתמודד עם בעיית ההזדקנות מהשורש. מה אנחנו סך הכל אומרים? שאם אנחנו נתערב באופן טיפולי לתהליך ההזדקנות, אנחנו נוכל למנוע כמה מחלות זקנה, להאריך תוחלת חיים בריאים. יש משהו יותר הגיוני מזה, שדרך יותר טובה להתמודד עם הנושא. לא.
לא שוב, אבל בגלל זה אני חוזרת לעניין המילים, כי כשאנשים מתבגרים באיזשהו שלב, אחרי גיל מסוים, הם כבר מתחילים להזדקן, לא כי הם נהיים ישר חולים, כי יש איזשהו גיל שבו אתה כבר מתחיל לחשוב על עצמך כזקן יותר. אז בגלל זה הם לא רואים בזה כמחלה, כי זה לא תמיד ישר בא עם מחלות, אבל כשעוברים את גיל ה-70, אפשר כבר להגיד שהזדקנתי, ולא אוטומטית לראות את זה כחליתי. אתה מבין? ועל זה שתניה
עוד הערה מהיציאה?
עוד הערה, כן.
כי אני חושב שניר משדר את המסר הנכון, אבל הוא נשמע מפחיד, כי בעצם הוא אומר הבעיה היא של כולם, ואז אפילו בקונטקסט של חברות הביטוח זה לא טריוויאלי. הנה הצעה שאפשר להתחיל איתה, והיא הצעה שאני חושב שהיא יותר נוח לאנשים. כל הדברים שמדברים על שעונים ביולוגיים, אני דיברתי אחד, אבל דיברו פה על עוד, הם כולם אומרים שלא מזדקנים באותו קצב, גם הנתונים של ניר אומרים את זה. אז אפילו בתור מטרה, בואו ננסה להאיט את הזקנה, זה אנשים שמזדקנים מהר. זה כבר אוכלוסייה יותר קטנה, אפשר לזהות אותם, כן? זה בתור סקאלה. אם נתחיל לעשות את זה בתור צעד, אחרי זה אפשר להמשיך. אני חושב שזה פשוט אותו מסר, אני מאוד מאמין במסר של ניר, אבל פשוט הוא נשמע נורא כאילו גדול ולא פתיר.
אז אתם יודעים, בואו נדבר רגע על ניצוצות של אולי פתרונות. ניר, אתה הזכרת, ציינת את הארבעה אחים האלה, שחיו עד גיל...
בין 102 ל-110.
ומהם אפשר היה למצוא, לא רק מהם, אבל אפשר למצוא, זה אוכלוסיית מענים, נכון? אנשים שעברו את גיל 100, ואפשר לראות שיש להם מפנה גנטי שמביא לאריכות חיים. אמרת שיש שתי תרופות כבר שאפשר לטפל באמצעותן. תספר יותר על התרופות האלה, כמה הן באמת נפוצות בשוק, ומי מקבל את הטיפול הזה.
בסדר,
ואיפה קונים את זה?
סליחה, אני אעשה לכם אותו דבר.
אוקיי, אתה שוב משנה את השאלה.
אני אספר על הפוליטים, אבל אני רוצה להגיד על מה ששי אמר. תראו, זה לא רק הזקנה. אנשים שמקבלים טיפול כימותרפי ורדיאשן תרפי לסרטן וכל הדברים האלה, הם מזדקנים בממוצע 10 שנים, הם 10 שנים מזדקנים אחרי הטיפולים האלה, כי מה שקורה להם זה חלק, מאיצים את חלק מהתהליכים הזקנה. אז יש את הקהל הזה. אנשים עם HIV, אני לא יודע כמה יש בארץ, יש המון בארצות הברית, הם גם מזדקנים יותר. נכים שהם על כיסא גלגלים, אז אין להם, הם לא עושים פעילות גופנית, והם גם משמיעים והכל, הם גם מזדקנים רק. אז אני מסכים שיש אנשים שיגיעו לגיל 100 בלי עזרה, אבל זה אפילו לא רק הזקנים, יש עוד הרבה אנשים שאנחנו נוכל לעזור להם לעשות את זה. עכשיו, איך אפשר לעזור? כמובן, דבר ראשון, בפעילות גופנית.
אפשר עוד הערה לסדר, בדיוק את מה שאתה רוצה, שתחבר את שניכם. ראשית כל אני מסכימה לגמרי איתך, ואני חושבת שכמו שאתה אמרת בלהט שלך, יש לנו מדינה נהדרת, ואני יכולה לתת רק דוגמה אחת, שישבו פה בארץ, מסינגפור ומארצות הברית ומיפן ומאוסטרליה בנושא של אייג'ינג, והם אמרו, אתם הדוגמה שלנו מערים חכמות שמטפלות בזקנים, ועד הרפואה שבאמת תזהה את הפרסונל פס של כל אחד. וזה מה שאנחנו צריכים להדגיש. ואני לגמרי אומרת לך שיש לנו פה את ה-competitive advantage, ויש לנו מדינה, ואפילו כמו שאני קראתי, זה הייתי קוראה לזה healthy aging, ואפילו לא GEROSCIENCE. healthy בכל שלב של הגיל שלך. זה בכלל לא משנה. וזה המטרה, ואנחנו יכולים לעשות את זה. ובאמת צריך לחבר את כל היכולות של כולם, ויש לנו את הדרכים. זה רק כאילו להוריד את המחיצות, ולהתחיל לשלם את זה.
אז עכשיו אני אענה לך על השאלה. דבר ראשון, לכולם, פעילות גופנית היא הדבר הכי חשוב שאנחנו יכולים לעשות כרגע. אז יש שתי תרופות שיש בהן שימוש נרחב, אז אנחנו יודעים שזה לא מסוכן, ואנחנו יודעים שזה מטפל במחלות. אחד, זה התרופה הזאת שקוראים לה מטפורמין או גלוטופאז', שהיא בשימוש נרחב גם בארץ. אני חושב שיש חולי סכרת שהם בערך בין 5-8% מהאוכלוסייה, והם בדרך כלל כולם מקבלים את התרופה הזאת. מה שהם לא יודעים זה שאפילו הם יותר שמנים ויותר חולים, ברגע שהם מקבלים את התרופה הזאת הם חיים יותר ארוכים, הם מתים פחות, ויש להם פחות סרטן, פחות מחלות לב, פחות האלצהיימר וכאלה. אני לא מציע לתת את התרופה הזאת כרגע, כי אנחנו רוצים להראות שהיא באמת עובדת בקונטקסט של הזקנה. תרופה שנייה, וזאת התרופה שיש לה את ההשפעה הכי
והפעילה, והעובדת, וכל הדברים האלה, ונכון, וכל
מיגור הזדקנות התאים. אולי נקרא לזה ככה.
מיגור הבעיות הרלוונטיות. כן.
מניעת ההזדקנות הניהולית והארכת תוחלת החיים הבריאים. זה הסלוגן, ובואו נעזור לו.
אבל צריך משהו יותר קאצ'י.
אנחנו לא משתמשים.
לא, אנטי אייג'ינג זה שפכטלים, זה מסכות ששמים בשביל להסתיר, זה לא בשביל...
עכשיו, אני רציתי רק להתייחס, כל מיני ביקורת נאמרה פה לגבי העובדה שנעשים מחקרים ואין ממש פעילות. זה תלוי את מי שואלים, ששואלים גם את משרד המדע שתפקידו לעשות מחקרים, וששואלים אותנו, כשאנחנו פה באים, אותי באופן ספציפי, לא את הרשות לחדשנות, אבל כשאנחנו מדברים על פיתוח טכנולוגיה ופיתוח ומדע, זה באמת זה דברים שבתהליך. אפשר להגיע לרמה מסוימת, שיש תהליכים בסופו של דבר, וזה צריך להיות במקביל. כלומר, צריך לעשות את מה שאפשר היום, וצריך להתקדם, וכל הזמן לשמור שיהיה בצינור משהו שמתקדם, גם באקדמיה וגם בתעשייה וגם בחברות תעשייתיות קטנות וגדולות, ולקדם את כל מה שאפשר בשביל שכל היוזמות יתחשבו בכיוון הזה וילכו לכיוון הזה ויסייעו לאוכלוסייה הזאת. באותה מידה, משרד הבריאות זה בסדר, אפשר לפנות למשרד לשוויון חברתי, ויש יוזמות ויש... זה לא סותר, זה ממש ממש לא סותר.
זאת אומרת, מה שאני אומרת זה מודעות. קודם כל בואו תשימו את זה על השולחן במודעות ובסדר עדיפויות נכון. זה כל מה שצריך היום. ואני באמת מסכימה, מיגור הזקנה זה לא רוצים למגר כלום. אנחנו רוצים להמשיך לחיות באיכות חיים ראויה כמה שאנחנו חיים. אפילו אם הייתי צריכה לדרג אישית, הייתי רוצה איכות חיים ואחרי זה אריכות חיים. קודם איכות, שאתה אזרח שמרגיש טוב עם עצמך כמו שהוא אמר, ושתורם לקהילה, ויש היום יוזמות מדהימות לתרומה ולתת לאנשים האלה איפשהו סיבה לחיות מאומנות ועד תרומות ועד ילדים. יש המון דברים, רק צריך לרצות לעשות, ויש לנו יותר מהרבה מדינות אחרות מבחינה חברתית את היכולת לעשות את זה.
אפשר הערה קטנה בתור גריאטר? כשאני סיפרתי לחבר'ה שלי בפקולטה לרפואה, כשסיימתי את חוק לימודיי, שאני רוצה להתמחות בגריאטריה, אתם יודעים מה הייתה התגובה שלהם? תגידי, זה לא מגעיל אותך לטפל באנשים האלה? כשאני הייתי בפנימית, אפרופו ברמב"ם, לא אצל אבא שלך, בפנימית השכנה, רופאה בכירה שמשמשת כמנהלת מחלקה, באחד מבתי החולים הגדולים ביותר, ליוותה אותי כמתמחה צעיר, בביקור שערכתי, והיו לי 5 פציינטים, 2 יחסית בגיל צעיר ו3 מבוגרים. אז תחילה התחלנו את הביקור עם 2 הצעירים יותר, וכשהתכוונו להגיע לאותם אנשים, 3 מבוגרים אמרה, ועכשיו שימו לב במשפט, אישה חובשת שביס, ועכשיו נלך לעשות רפואת גן חיות. המשפט הזה נצרב בתודעה שלי. נצרב ואומר...
עכשיו נלך לעשות רפואת גן חיות?
גן חיות, ומדובר באישה דתייה חובשת שביס, התחנכה ברכי, קבל את אביך בקמך, למען יקום ימיך. זאת אומרת שאנחנו קודם כל צריכים לרפא את החברה שלנו. בתפיסה שלה, לאדם המבוגר, שהוא אדם בכל דבר ועניין, גיל זה לא מדד. גיל זה לא מדד.
אז אני מציע לקרוא לזה שדרוג הזקנה.
או, שדרוג הזקנה.
אני חושב שאני מניח שאנחנו הולכים לקראת סיום. אז אני חושב שצריך לצאת מאיזושהי קריאה מהכנס הזה. ואני רוצה להציע, כן? ואני מציע למשרד הבריאות, שלאור כל התוכניות הלאומיות שהוא מכין ועושה, הוא היה צריך לשים בראש היעדים שלו דבר אחד מאוד ברור. היום אנחנו יודעים שהקצב של העלייה בתוחלת החיים מלידה גדולה מקצב העלייה של תוחלת החיים בבריאות טובה. הוא צריך לשים את היעד המרכזי להגדיל את איכות החיים בבריאות טובה. זה צריך להיות היעד של משרד הבריאות, ואני חושב שזה הלקח בעצם מכל הכנס הזה.
כי היום הרבה אנשים שאתה אומר להם עד 120, אומרים במצב הקיים עד 120 זה כללה, זה לא ברכה.
אז אני אומר, מבחינת משרד הבריאות, זה צריך להיות היעד שלו. וזה צריך גם להיות יעד לאומי. וזה צריך לשלב גם את כל המערכות האחרות. לכן אני גם מאמין גדול בעניין של תוכנית אב לאומית. כי זה קשור גם לדברים, אבל זה לא עומד בפני עצמו.
אז איליה, אני רוצה שאלה אחרונה לך כאקטיביסט שדוחף ומנסה להעלות מודעות לעניין הזה. מה הקשיים, מה השאלות או מה הזלזול אולי שאתה נתקל בו כשאתה מנסה לקדם את העניין הזה?
טוב, שוב, מה שאנחנו מציעים זה אולי הדרך ההגיונית ביותר והסבירה ביותר והמוצדקת ביותר מבחינה מדעית להתמודד עם אתגר ההזדקנות, כן? והבעיה שאנחנו נתקלים בה שאנשים לא תמיד מתנהגים בהיגיון, ואפילו אם הם מבינים את ההיגיון, הם לא תמיד פועלים לפי ההיגיון, אז אני כאילו...
אבל זה מעניין ההסברים לכך שאנשים פועלים בצורה לא הגיונית, כמו רובנו פועלים בהרבה דברים בצורה לא הגיונית. מה גורם לנו לפעול בצורה לא הגיונית בעניין הזה?
זה כבר משהו עמוק בטבע האנושי, למה אנחנו פועלים בהיגיון, אבל דווקא בנושא הזה אנחנו פשוט צריכים ללמוד את ההיגיון, להבין אותו קודם כל ולפעול לפי ההיגיון, שזה אומר באמת לשים את המשימה הזאת כיעד לאומי, בעדיפות לאומית ראשונה, ולהתחיל להשקיע משאבים בנושא האלה, להתחיל לפתח מדיניות, דרך תוכניות ממשלתיות.
אבל מה אתה שומע מאנשים שאתה מדבר איתם על זה, וגם אם אתה יכול לספר לנו באמת על יוזמות בינלאומיות, כלומר, מה האתגרים שאתה נתקל בהם כשאתה מנסה לקדם את הנושא הזה?
מה שאני שומע, שאתה צודק, נכון, זה מה שצריך לעשות, אבל אנחנו לא נעשה כלום, כי יש לנו סדרי עדיפויות אחרים. אז כן, אז אנחנו באמת צריכים להבין שזה מה שצריך לעשות, ולעשות את זה.
חן, אני רוצה לומר משהו. אני אזרח ותיק, אבל אני גם יזם, ויש לי מיזם שאני הולך להתחרות עליו בכנס ריבוט, אני לא יודע אם את מכירה את הפורום הזה. זה פורום של ארגוני הבריאות, וב-24/9 יש לו את הכנס השנתי, ויש שם גם תחרות על מיזם הבריאות של השנה. והמיזם שלי נקרא בריאות עד 120. אוקיי? זה מאחד את שני הדברים. ושם אני הולך לדבר על מודל כלכלי, שמאפשר לטפל בכל האתגרים האלה, מבלי להגדיל את התקציב. זאת אומרת, זה מודל שפונה, הוא ברח, זה התקציבן מהאוצר, שאחראי הרפרנט של הבריאות. אבל זה מודל שמאפשר בעצם בצורה של דיבידנד, longevity dividend, על כל חיסכון שחסכנו בשנת מחלה בגלל שהארכנו את גיל הבריאות, לקחת קופון לטובת מניעה וחינוך. וחינוך מתחיל בגיל הרך, לא שמבוגרים. כי אנחנו יודעים כבר שילדים יודעים לחפור להורים ולהשיג מה שהם רוצים, אז אם הם ירצו בריאות, ההורים גם יהיו בריאים. אתה תעזור לי, יצחק, מה זאת אומרת? גויסת.
צריכים לסיים, אבל אני חושבת שזה היה דיון ער, עם הרבה השתתפות מהקהל, וטוב שכך, ועד 120.